Hold on, I'm just getting, hold on, I'm just getting the slide set up.
No problem. And just for everyone, please, if you have questions or comments, put them in the chat. We'll moderate it so that we can bring up some questions at the end too.
I am, my Zoom just suddenly went into minimized view.
Do you want Tiffany to pull up the slides and have her advance for you?
Let me try one more thing.
We've only had 15 months to practice with Zoom.
I know that's what's killing me about this. It went to the minimized view as soon as here. Let me hold on one moment while I try to reconnect here.
Okay.
Actually, I apologize. We will make up the time, and here we go. Okay. Tell me if you can see my slides.
Yes, they're up.
Okay. All right. Thank you for bearing with me through that tech problem. Colleague Sean, Tunis, and we're going to talk about one of the project areas that and a couple of things to start off with. Number one, it's great to talk to the
Joe, you cut in and out, and then you went into.
I just got a message that I was muted, but
Yeah.
Can you hear me now?
Yes.
Okay. Sorry about that. We'll get this back. So two things. One is it's really great to be at the Yale-Mayo CERSI. The CERSI programs, including the one at Yale-Mayo, are a fantastic way for FDA to interact with researchers at institutions around the country. And in particular, we have a great project with Yale-Mayo in the real world data space. And it's fantastic to see that a number of the presentations today also have topics that are, I think, going to be relevant to some of the things that Sean and I talk about. The other thing is that I was a PhD student not so long ago, or I guess it's increasingly long time ago, at Yale, cell biology. So it's nice to be back, even virtually only. So we're going to talk about real world data and real world evidence used to inform FDA's COVID-19 response. And this is going to be a short overview. Obviously, we only have about 20 minutes. We want to leave time for questions. So I'm going to run through this fairly quickly, but we'll give you a taste of not only the projects that we've been working on, but the why behind some of these projects, and crucially, the challenges that are outstanding. It's worth noting that real-world data, real-world evidence has been something that not only you all in the Yale CERSI context and more broadly in your research, many of you in your research have been working on, but FDA has been working on this from the scientific and policy perspective for many years, and in that policy development, scientific development at the agency was punctuated especially by the passage of the 21st Century Cures Act in 2016. And, largely, especially in my work pre-COVID and the work of many of my colleagues at FDA and outside of FDA, the real-world data and real-world evidence was focused on developing evidence to support review of product applications. Extremely important area, but only one area that we think data can be important to inform FDA's decision-making. And it really, in my mind, took COVID and the stress of COVID on our public health agencies, including FDA, to reveal many of the ways data is needed to inform the types of decision-making in the pandemic response and we think in the future. So just to be concrete about it, clearly real-world data can inform FDA's response to COVID-19. Sean is going to talk a little bit more about some concrete areas that we've been looking at and specific real-world data projects, as I mentioned. But when you think about it generally, real-world data obviously can provide updated near real-time insights into the clinical experience of patients. And we're not looking just at outcomes. We can look at treatment patterns. Also, we can look at the patterns of use of diagnostics and vaccines using data from claims, ChargeMaster, EHR data, other types of real-world data. We can use data that helps us understand the evolving course of the disease. If you recall, a year ago, one of the most crucial areas for everyone who's working on COVID was to understand the disease as quickly as possible. And obviously, we're still in the process of doing that a year later, more than a year later. Real-world data allows for analysis of outcomes that may not be feasible to evaluate in clinical or traditional trials. This is true, obviously, outside the COVID context. So what are the applications for FDA? Our projects are focused on developing scientific and technical tools to enable analysis and identify gaps and providing continuous updates to inform our response. I'm going to focus on the first of these, and I think that's the most important component of the projects we're talking about is how do we use projects in the COVID context to learn how to use data. So,
hey, Joe.
Yes.
I'm sorry to interrupt. Your slides, when you originally muted, dropped. So if you're walking through slides, can you pull them back up?
Absolutely. All right. We will get this presentation going. Now.
Yes, we can see them.
All right, thanks. It's an honor to be here at Yale, and thank you for bearing with me through the presentation difficulties. So I wanted to focus on a few questions, just to ground everyone in the projects that we're working on. In our group at FDA and across the agency, it's important to think about what are the types of decisions FDA needs to make in a pandemic. I think the most common type of decision that we think about in the larger community are the regulatory decisions. FDA is a regulatory agency. We make regulatory decisions, including decisions on issuing an emergency use authorization for a drug to treat COVID-19 or a diagnostic or a vaccine for COVID-19. But there are many other decisions that FDA makes, including operational decisions, how to prioritize its work that when you're thinking about the rapidly evolving pandemic are in many respects as important sometimes as the regulatory decisions. And then obviously it's important to think about what types of decisions could benefit the most from better information. I think the COVID-19, if nothing else, I think we can all agree that it has made us as a community, as a public health community, think more clearly about what it means for questions and decisions to be science-based. And so we are thinking about the types of decisions that can benefit the most from better information. I think that when we think about better information or data informing decisions that FDA makes, what are some of the challenges? If we want better information, we want information more quickly to inform decisions. What are some of the challenges? Data infrastructure, connecting the data. These are areas that Sean will talk about. It's crucial in our project so that we've learned over time to understand the data and how to analyze data. Sean will talk through some aspects of our projects that have focused on these issues. And an area that I think is extremely important that we won't talk about today, but I think is the is worth additional conversation, is learning how to make decisions using information that's incremental and non-traditional. And this is one of the most important themes to me in the context of real-world data, because we're talking oftentimes about novel data sources that contribute information that's more incremental than some of the more traditional data sources that we've relied on at FDA, namely RCTs. So Sean, if you're online, I'll kick it over to you to move on to the next slide.
Great. So just checking, can you all hear me?
Yes, we can hear you, Sean.
Hello. Okay, great. And good. And hopefully we're on the slide that's just got the title Real World Data. And Joe, thanks for advancing the slides for me. I am sitting in a parking lot outside a COVID vaccination clinic, which I signed up to do today, forgetting that I was giving this presentation. So I'm comfortable in my car, but hopefully it's not going to pose any technical problems. So just a kind of a quick review, and I know for some of you, some of this is going to be too basic, and then it's going to quickly get too complex, but hopefully we'll convey some of the key points. I appreciate Joe Franklin kind of setting up the kind of the FDA policy context and some of the things related to real-world data that the agency was hoping to be able to efficiently learn through real-world data. And just to get us started on sort of some common definitions. So when we talk about real-world data, it's essentially the universe of health data that is collected everywhere except from traditional clinical trials, or at least that's, you know, widely adopted definition. And as this figure shows, real-world data comes from many, many different sources, including electronic medical records, claims data from payer claims data, wearable devices, laboratory reports, you know, data from hospitals, et cetera, et cetera. So this is probably not a complete representation, but gives you some idea about the kinds of sources of real-world data that we've endeavored to use to inform many aspects of our COVID response. Real-world evidence is simply the clinical evidence that is derived from the analysis of real-world data. So if you could go on to the next slide, that has the title, The Healthcare Data Ecosystem. As you can see, this is a huge collection of company logos. Must have been some graduate student summer project or a young consultant at DataVan. And you know, you will share the slides. You may be interested in pouring this over, but it's really, the slide is meant to convey that one of the major challenges that you encounter when you try to make sense of real-world data is that the ecosystem of health data is large, extremely complex, and expanding rapidly. In some sense, that's good news and bad news. The good news is the quality and the volume of the data is, you know, in a kind of an exponential growth phase, and it is the quality and volume of data is probably growing much faster than our ability to use it intelligently. But that's, you know, that again, that's you know, a great opportunity. So this compilation of the companies that are involved in the health data ecosystem, just a brief, you know, kind of introduction to this figure. The column on the left illustrates the various originators of real-world data. So the electronic health record companies, pharmacies, laboratories, wearables, et cetera. The column on the right is organized by real-world data services, kind of separated by the kind of the customers. So those being, you know, healthcare providers, risk-bearing organizations like insurance companies, life sciences companies. So this is a kind of a compilation of all the organizations that are providing real-world data and analytic services to those organizations. And the middle represents some but not all of the major health data aggregators. So as you can imagine, this figure reflects enormous investments in people's time and expertise and money, and perhaps, you know, if nothing else, suggests the enormous expected value that people see in, you know, being able to derive useful insights about healthcare delivery as well as the risks and benefits of medical products, you know, that could be derived from being able to use this real world data. So if you can go on to the next slide, it's titled Real World Data for COVID-19. So this is just a brief introduction to the Reagan-Udall Foundation Evidence Accelerator, which was set up to essentially help FDA and others make sense of this complex ecosystem of health data. The FDA worked in partnership with the Reagan-Udall Foundation to stand up what is called the COVID-19 Evidence Accelerator. The Reagan-Udall Foundation, as some of you may know, was established by Congress in 2007 as a partnership for the FDA to assist the FDA in advancing regulatory science. This program of the Reagan-Udall Foundation, the COVID-19 Evidence Accelerator, provides a forum where experts and stakeholders can work with the FDA as well as other government agencies, the data and technology community reflected on the previous slide, to, you know, share insights, collaborate, develop methods, and essentially, you know, an opportunity to work together in a sustained way to figure out how to make sense of real world data in ways that are useful to the regulatory responsibilities of the FDA. The work has been conducted in a real open and sharing collaborative approach. So, you know, really something that, you know, the FDA has limited ability to do on its own, but an organization like the Reagan-Udall Foundation serving as a host really allows all of these disparate organizations and experts to kind of team up and share their learnings and make things go much faster. And the primary goal for the Evidence Accelerator is to gain a deeper understanding of how to rapidly leverage this health data ecosystem to augment our understanding of COVID-19, improve public health, improve regulatory decision making. If you could go on to the next slide with the rocket ship. It's kind of a 1950s rocket ship, but that's at least it makes a nice image. And this is just a kind of a timeline of some of the activities that have been conducted under the, you know, through the forum of the Evidence Accelerator. It became operational fairly early in the pandemic, launching a series of meetings and projects beginning in April of 2020, with a focus initially on therapeutics. Then, in order to support work on COVID diagnostic tests, the Evidence Accelerator expanded its work portfolio in May of 2020, and then extended to doing work on vaccines starting in March of 2021, you know, approximately timed with the commercial availability of the initial COVID vaccines. And to this point, the Evidence Accelerator has convened well over 60, probably more in the range of 80 virtual meetings. Initially, they were weekly. Now they're bi-weekly. Several hundred participants over that period of time. And a lot of useful work has gone on with several specific targeted projects, a couple of which I'm going to talk about in more detail, you know, in the remaining minutes of this conversation. So the next slide, this is just sort of a schematic of what we refer to as a parallel analysis project, which is one of the major activities that is conducted under the Evidence Accelerator. Basically, in a parallel analysis project, you have multiple data holders working together to address a single high priority research question. And working together involves identifying, you know, and refining what exactly the question is, what are the key data elements needed to address that question, how does one define similar patient cohorts across the different data sets using the available information, diagnostic codes, as well as hospital admission codes, et cetera, diagnostic test results, and then how to measure outcomes. So all of this is then kind of converted into what's referred to as a mass standardized analytic framework. And as you can see in this schematic, you've got 4 different data sources. They're conducting separate analysis using this common study protocol or master protocol, and then producing towards the bottom, from each data source in the parallel analysis are emerging the results reflecting the findings of that data source. And then a summary analysis is done by comparing and contrasting the results that are derived from each of those individual data sources. So if you can go to the next slide, I'm going to go through kind of at a high level one of the initial parallel analyses that was done, which has now been published. And this one was focusing on the use of hydroxychloroquine and azithromycin in patients who are hospitalized with a diagnosis of COVID-19. For the sake of time, I won't go through each of the individual questions, but in general, the point of this parallel analysis was to characterize what the patient populations look like, to characterize the timing of the therapy in the illness trajectory, and then look for safety signals and compare clinical outcomes in patients treated with hydroxychloroquine, azithromycin, both, or neither therapy. As some of you, or probably most of you know, this was an area of kind of extreme clinical, scientific, and political controversy related, as folks early in the pandemic were looking for, any signals that there might be some therapeutic benefit or potential risks to these and a number of other therapies. So if you can go to the next slide with the title, Total Population.
And Sean,
Yeah.
I was going to mention and emphasize that point. I think that, we're reporting some of this information many months later, but in my mind, I'm wondering, what value in early 2021 was it in looking at hydroxychloroquine? But to emphasize the point, very early on in the pandemic, what we wanted to do is set up these processes and parallel analysis structure so that we could start evaluating the data that was available. And at that time, the data available was patients with or without hydroxychloroquine. So it was an exercise in developing this, answering the questions that we could at the time so that we'd be prepared for more complex data projects as the pandemic evolved. Back to you, Sean.
Yeah. No, I think that's a really helpful insight. And in fact, we'll kind of come back to that, I think, in the summary shortly. So just to kind of finish out this kind of case study, if you look at the slide, hopefully the one that's showing on total population times treatment, this is a slide that shows the number of patients from each of the 6 real-world data sources that participated in this particular parallel analysis. So from Health Catalyst, there were over 7,000 patients hospitalized with a diagnosis of COVID-19 that were then distributed across these various treatment groups, and you can see the 5 other data sources and the number of patients that were contributed, by that data source. And then just to jump to the results in this is titled Adjusted Directional Outcomes. These are the survival results. There's also results on safety, and then a number of results just describing the patterns of care, the demographic characteristics of patients in these different treatment groups. And as you can see in these results from the group 2, or the second of the databases, you can see that kind of color-coded here that the patients treated with a combination of hydroxychloroquine, azithromycin, or hydroxychloroquine alone had survival outcomes that were approximately the same as the control groups from this database. Similar results from group 5, database 5, and then in the third one, the results for the azithromycin patients were similar to control, but for either of the hydroxychloroquine alone or in combination with azithromycin had survival that was worse than the control. And again, kind of going back to Joe's points, in some ways, the results themselves, while interesting and needing to be kind of looked at in the context of all the other results that were coming out, looking at these therapeutic interventions and their outcomes, what was really most important from this exercise is a very accelerated process for learning about how to use these various rich databases and the ability to conduct the same analysis across multiple real-world data sources, and then from that gain insights into what was hard, what was difficult, how to improve, and essentially how to improve for the future. I'm realizing that we're kind of getting to the end of the allotted time and wanted to leave a little bit of time for questions. I think I will kind of skip over, but just introduce you to the work that we did on the diagnostic side. So there's a figure here, the first figure on this, evaluating COVID-19 diagnostic test performance. Essentially what we set out to do here was to see if we could use real-world data to identify patients who had a positive COVID nuclear test, a PCR test. For those patients, identify what their results were on serology tests at some time later, an antibody test, with the expectation that most patients who had a positive PCR test should at some point later have a positive antibody test, and then ideally then looking at the long-term outcomes for those patients, including future hospitalizations, clinic visits, etc. And again, this exercise was set up mainly to explore how whether and how we could actually piece together data sets that would provide this information, in the hopes then that could be used in a repeated fashion to look at the real-world diagnostic test performance of the hundreds of COVID diagnostic tests that were approved through emergency use authorizations and therefore subjected to relatively less rigorous clinical evaluation or clinical studies prior to them being made available for use in patient care. So I think unfortunately we're going to have to skip over the elegant story about Heidi and her diagnostic test results, that we can come back. Maybe you'll invite us back another day to go through that detailed discussion. But I want to kind of jump to some of these lessons learned, which I think Joe has already touched on. So the final slide, and just to kind of re-emphasize some points we've made in this discussion. There's an enormous and growing amount of real-world data. Again, thinking back to that health data ecosystem slide, turning real-world data into real-world evidence is far from a simple task, but an enormously compelling and important task. As we've discovered through the work of the Evidence Accelerator and a large hundreds of talented collaborators, the best way forward is somewhat by trial and error and identifying important questions, making the effort to generate data to answer those questions, and through that learning more about what you can and can't do with real world data. And the best way to do this work is through collaborative communities like the Evidence Accelerator. And COVID-19 provided a profoundly intense motivation to focus on these challenges in a sustained way, and I think has kind of in an exponential fashion advanced the understanding, not just at the FDA, but in this entire community of real-world data experts and real-world data providers in, you know, setting us up, I think, in the future to really be able to conduct this kind of work in a much more informed way going forward. And maybe throw it back to you, Joe Franklin, to see if there's anything you wanted to add in terms of lessons learned as we, you know, round this out.
No, thanks, Sean. Yeah, I think that this is as you can tell, a work in progress at FDA and in the broader community. I know many of you are involved in understanding how to use data better in the COVID context and beyond. I'd like to think that at FDA we could come back after some time has elapsed and say that not only are we ready to use data more quickly because of in the next pandemic or public health emergency, but because of what we learned during COVID-19, but broadly, constantly to improve the agency's ability to respond and perform its mission. So that's really fantastic to be able to talk to you all today. Sorry again for the tech glitches at the beginning, but looking forward to the other presentations during the seminar. Thanks.
Thank you, Joe, and thank you, Sean. I mean, it's really incredible sort of what's happened at the agency, you know, and obviously COVID prompted a lot of that, but there was obviously a runway that had been sort of built up. But the Evidence Accelerator certainly did accelerate things in terms of bringing groups together and working towards common path. I guess one question I have for you guys is, you know, it's great for FDA to sort of independently or working with these collaborators to evaluate these therapies now, like in the context of, you know, hydroxychloroquine after the emergency youth authorization. But what is the decision making, you know, when a company is not bringing it to you guys, asking for approval, or you know, you guys are now sort of leading these analyses that are demonstrating that the medication isn't working any better. You know, it's like a funny twist on the, well, you know, how are these decisions made? I don't know if you want to comment on that and some of the challenges you face.
Yes, yeah, and this is a fantastic question, Joe. It's a sensitive topic because, you know, obviously FDA has been under the microscope with respect to decision making around EUAs from the very beginning, as is appropriate given the importance of the agency's decision making. So the pandemic. So I think that one reaction or reflection that I'll have based on your question is that the types of decision making during the pandemic is different in a lot of ways compared to the normal course of decision making at FDA. One of the reasons I believe is because is that the agency does need to take some more proactive decisions quickly and not rely as much on the generation of evidence from an applicant a manufacturer who's applying to get their drug approved by FDA, and that teaches us I think to look for other places that the agency works on, other decisions that the agency makes within the pandemic context and outside the pandemic context. So not just EUAs, not just pandemic. Where those decisions can be informed by data and information that the agency itself generates, rather than situations where the agency is waiting for a manufacturer to submit the data. The dividends around this kind of work also pay off in the more traditional application review context, because we're getting more sophisticated, we're getting better through these projects at evaluating novel sources of data so that we're going to be able to apply that expertise when reviewing more traditional FDA regulatory actions or items like new drug applications.
There's one question in the chat, it's multiple parts, but I'll synthesize to it just a couple points. One is, how are data collected on vaccine administration through all these different data sources when it's not within a health system using EPIC or Cerner? And the sort of leeway, you know, steps into the question of, like, you know, in what year or how many years do you think it's going to take to smooth all this out? And I guess applied into non-COVID spaces.
Right. Well, so I'll hit the first question which is that real data collection on vaccines is a huge priority. It's something that the Center for Biologics, CBER at FDA is focused on. And there are challenges, including in the evidence accelerator this week around particularly getting useful information related to the vaccination status of records in our sources of real world data. And I think that's something that FDA is working on building, not only building on what it has now, the surveillance capabilities that it has now, so that there are more real-time information, I think, for the future, especially around vaccines, given how important that's become over the past year. And then the second question, so remind me, sorry.
Well, I asked the impossible to answer question of when we'll be able to do this
reliably. I don't know, when do you think, Joe? I
don't know. We got to get all the health systems to adopt a common data model and submit everything and make it, get UDIs embedded too, so we can do medical device. There are a lot of steps ahead.
There are a lot of steps. I think we've learned quickly in the past year, and I say we, I mean like the broader community. And I think that FDA's participation in that is important. And I think, to Sean's point, we've learned that we need to work together in new ways. It sounds overly general, but it reflects the fact that data exists in a lot of different ways in a lot of different places. And until you can get it together and get it analyzed by people who understand the underlying data, when there isn't a common data model, it's hard to get useful information out of it. And then the one other piece that I'll say that I alluded to, but that we obviously didn't have time to talk about this morning, is that we've got to figure out better ways, including at FDA, and maybe especially at FDA, of learning to make decisions, even relatively small decisions, based on novel data sources. And that's a big part of this process on the demand side, not just creating new information, but learning how to use it.
Thanks, Joe. With that, I want to thank you and thank Sean for joining us today. I mean, it's really important for our scholars to hear about the high-level, high-quality science that's being done at FDA to inform decision-making. I hope they found it interesting and it's inspirational. You guys are welcome to stay with us, join, ask questions, along with so many others who are here with us from FDA. I'm going to turn it over to my colleague, Nilay Shah, over at the Mayo Clinic, and I think we'll skip a break and we'll just go right into the first set of presentations.
Great, thanks, Joe, and thanks again to Joe and Sean for joining us and just a really insightful presentation. With that, we'll move into our first session of the day, where we'll have 3 presentations, and then after that, we'll have some time for Q&A. Across those 3 presentations. The first presentation will be from Victoria Bartlett from Yale University. Her presentation is titled Physical Activity, Patient-Reported Symptoms, and Clinical Events, Insights into Post-Procedural Recovery from Personal Digital Devices. Welcome, Victoria.
Thank you. So as Dr. Shah said, my name is Vicky Bartlett. I'm a medical student at Yale, and I'll be talking today about my project, Physical Activity, Patient-Reported Symptoms, and Clinical Events, Insights into Post-Procedural Recovery from Personal Digital Devices. So, sorry, I can't actually see. Okay, great. So next slide. Personal devices or wearables have been growing in availability and popularity in recent years. People can use them to track not only step count, but heart rate, heart rhythm, blood pressure, and now even blood oxygen saturation. And with the growth of these capabilities, there's also been growing interest in using these devices to better understand patient experiences outside of the clinical setting in people's regular daily lives. Next slide. One important outside hospital situation to understand better is post-procedural recovery. Now, prior studies have looked at characterizing post-procedural recovery using different real-world data sources. However, often these sources are siloed, and this prevents the aggregation of multiple types of data, such as subjective patient-reported outcomes and clinical events from the EHR, which could give us an even richer understanding of post-procedural recovery. Next slide. So one solution to this problem is Hugo, which is a novel mobile health technology that allows patients to sync their health data all in one place. Patients can connect different health records from multiple health systems, as well as their personal digital device data to Hugo. Moreover, when it's used in research studies, Hugo can also send surveys to participants to regularly characterize their symptoms and aggregate this with the rest of their health data. Next slide. So with Hugo's capabilities, we conducted an 8-week cohort study at Yale and Mayo, tracking 60 patients after they underwent catheter ablation for atrial fibrillation or bariatric surgery. In this paper published last year, the team demonstrated that the feasibility of using the Hugo platform to aggregate personal digital device data, patient-reported outcomes data, and EHR data. So the aim of this follow-on project was to analyze the data that was collected in the cohort study. Specifically, we wanted to study the association of activity with patient-reported symptoms. And in catheter ablation patients, we analyzed the associations of activity and symptoms with objective rhythm assessments using a mobile EKG device. So in order to do this, we analyzed step count and heart rhythm data, subjective reports of pain and palpitations, and data on post-procedural healthcare encounters and medication changes in order to characterize recovery after catheter ablation and bariatric surgery. So in the 8-week cohort study, we analyzed the first 5 weeks of post-procedure data. This was limited by the patient-reported outcome measures, or PROMs, that we specifically used for this analysis. We only collected the first 5 weeks of data for that. We included 30 atrial fibrillation ablation patients, 29 bariatric surgery patients, and all data was measured weekly. And any occurrence of the outcomes in a week accounted for the whole week. So we asked patients to use a Fitbit to record daily steps. We calculated median daily step count both weekly and overall for the whole 5 weeks. We also collected PROMs, patient-reported outcome measures, every week. And we asked patients to report whether or not they had pain, and we asked ablation patients to report whether or not they had palpitations. And lastly, we also asked ablation patients to use a cardiomobile device to make at least one heart rhythm recording per week, but they could make, on top of that, as many as they wanted. And we calculated the number of atrial fibrillation recordings. We also did some analyses of EHR as data of ED but so our outcome, we overall observed once during the 5-week period. And then we also looked week by week and evaluated the mean marginal effect of pain, palpitations, and AF episodes on median daily step count for a week. And we used generalized estimating equations, which allowed us to account for intra-subject correlation of step counts and observations of each of the variables. So when we looked at pain, across the 5 weeks, 30 patients reported at least one episode of pain, 16 patients reported no pain. And we found no statistically significant difference in median daily steps overall between these two groups of patients. Next slide. When we looked week by week, we also found, oh, sorry, one slide back. Yeah, great, thank you. When we looked week by week, we also found no significant marginal effect of pain on median daily step count. But you'll notice that we did find that each post-procedural week had an increasing mean marginal effect on step count as compared to post-procedure week 1. And we found the same result for the other outcomes that I'll talk about in a few slides. And next slide. Pain also evaluated on ED, we found effects. Next slide. For ablation patients, for ablation patients, 18 of them reported palpitations at least once over the 5 weeks, and 3 reported no palpitations. We found that those that did report palpitations had a significantly lower median daily step count overall than those that did not report palpitations. Next slide. Additionally, when we looked week by week, palpitations in that case actually did not have a significant marginal effect on step count. And next slide. And then when we looked at each individual post-procedure week, again, we found no significant effect of palpitations in any of the 5 post-procedural weeks. Lastly, for the ablation patients, we also looked at AF episodes recorded by the mobile EKG device. 15 patients recorded at least 1 episode of atrial fibrillation across the 5 weeks, and then 6 patients used the cardiomobile device but didn't record any AF episodes. And we didn't see any difference in overall median daily steps between these 2 groups. Next slide. Then when we look week by week and also took into account the number of episodes, we did an episode on median daily steps for that week. Next slide. And then you can also see when we look week by week, and each AF episode recorded in weeks 1, 4, and 5 had a significant negative mean marginal effect on step count. AF episodes recorded in week 2 and 3 also had a negative mean marginal effect on step count, but it wasn't statistically significant. So our findings show that real-world data collected from personal digital devices can offer insights into post-procedural recovery. In particular, we found associations between objective measures of activity and atrial fibrillation burden. Additionally, longitudinal intra-individual trajectories may tell us more than broad comparisons between patients. So in terms of future directions, next slide. Thanks. Future directions. So our study was small, and in the future, larger studies with more granular data will help us understand recovery better. Moreover, larger studies that look into patient outcomes could help us understand how these real-world data could guide clinical practice in the future. Some examples of potential applications are the integration of activity data with atrial fibrillation burden data could help inform post-ablation rhythm control interventions. And personal digital device data could also help to risk stratify patients after procedures for in-person versus telehealth follow-up. Lastly, patient-reported outcome measures data linked to medical device data could also help inform real-world device evaluation. Thanks.
Thank you, Vicki. That was a great presentation. We will take questions at the end of the 3 presentations. So with that, we'll move to the second presentation in this session from Yale University. And his title of his presentation is on reimagining real-world clinical trials to increase participation of African Americans for patients, sponsors, and regulators. Welcome, Christopher.
Thank you, Dr. Shaw. Can you all hear me? Yep, thumbs up.
It's a little bit louder would be great. It just is a little soft. But.
Okay, great. So my name is Dr. Christopher Cross. I'm a CERSI scholar, the founder and managing director of Cross River Strategies, a health equity firm committed to increasing positive health outcomes for underrepresented and vulnerable populations across the healthcare landscape, with a focus on life sciences. And I'm also a PI in Yale's Center for Community Engagement and Health Equity, and a postdoc fellow in the School of Public Health in the lab of Dr. Jeffrey Townsend. And as Dr. Shaw mentioned, the title of my talk is Reimagining Real-World Clinical Trials to Increase Participation of African Americans for Patients, Sponsors, and Regulators. Next slide, please. So let's unpack the title of the presentation. What is a real-world clinical trial? It's a term used to differentiate from traditional clinical trials as trials that include real-world data and real-world evidence, as we heard earlier from our speakers from FDA. And so what is real-world data and real-world evidence? And so the concept of real-world data and real-world evidence were introduced in the 21st Century Cures Act, which was passed in 2016. And since Congress and FDA have expanded further to define them as follows, which you can see on the slide. So if you think about the diagram that's there, we've got the cloud with the data. And so this is helping us understand what is the relationship to having a real-world clinical trial. So we start with real-world data. And so again, that's defined as data relating to patient health status and or the delivery of healthcare routinely collected from a variety of sources. And so what are these sources? So we've talked about those earlier today as well. They can include billing claims, electronic health records, registries, device-generated data, patient-generated health data, and diagnostic imaging. And so those can all then be leveraged to create clinical evidence. And so real-world evidence is the clinical evidence regarding the usage and potential benefits or risks of a medical product derived from analysis of real-world data. And so on the upper left is a screenshot of the publications and guidances about real-world evidence from the FDA's website. And they serve as a primary source to better understand real-world data, real-world evidence, and real-world clinical trials. Let me go to the next slide, please. So this slide depicts a very high-level overview of the key stakeholders involved in conducting a clinical trial: the patients, sponsors, and the regulators. And the typical flow of interaction of these stakeholders stems from an unmet need that's typically defined by researchers, scientists, and public health experts, which is then taken on by sponsor or drug developer, and then they develop a plan informed by the FDA guidances, publications, and Code of Federal Regulations. And when given approval to begin a clinical trial, they start to recruit and enroll patients, collecting samples and data through a randomized clinical trial or observation study through a series of phases. When submitting an application of their product for approval, where throughout the process, FDA regulators evaluate the sponsor's work, make sure the end product is safe and efficacious before reaching the public. And so you can understand how that dynamic works based on the diagram that's on the slide. And so typically that interaction from patients to sponsors is somewhat unidirectional, and then sponsors and regulators typically have this back and forth. But there are exceptions in the case where patients can directly interact with regulators. And so importantly, let me go back. Okay. And so notably, African Americans and other represented groups are not typically part of trial enrollees, and 80 to 90% are usually white Americans. And so this fact underlies the need for this talk to have an intentional effort on increasing enrollment of African Americans, that continually are disproportionately affected by many diseases, including COVID and colorectal cancer, to list a couple. Okay, next slide, please. So now that we have a general understanding of real-world data, real-world evidence, clinical trials, and its key stakeholders, let's understand how real-world data can alter the traditional clinical trial pipeline. So in an effort to stay on time and grasp the concept, I focused this presentation on the patient-generated health data within real-world data. And so what is patient-generated health data? It is health-related data created, recorded, or gathered by or from patients to help address a health concern. And so per the diagram, you can see how PGHD is leveraged across the product life cycle to include both pre and post market. And so if you look at the base on the graph, you can see in this gray bar around how can PGHD and really real world data in general be leveraged in these particular buckets in the pre-market and post-market spaces. Next slide, please. And so my CERSI sponsored project specifically explored the role of PGHD of African Americans. And so I was successfully able to partner and speak with Dr. Elizabeth Ofili and Dr. Priscilla Primu, who are at the Morehouse School of Medicine. And so they created an app called Health 360X. And so the data on Health 360X is directly from patients and users, excuse me. So this is real world data. And the app has over 1,900 people who have accounts on the app, and is capable of generating anonymized data, particularly input and engagement, noting that age, location, and race are not required fields. And so that said, this diagram shows how real-world data, and specifically PGHD, is captured through their app, stored in the systems, which has been successfully used to increase outcomes for patients with chronic conditions. And so if you look at the bullets there on the right, these are self-reported data from African Americans living with chronic health conditions. The app usage has showed better health management, and they actually conducted an internal study to compare the medical coaches versus familial coaches. And that showed that there was parity in achieving those increased health outcomes. And so I developed the research questions, as you can see below, to better guide our ongoing research to ultimately answer how can sponsors be intentional on including African American and other represented groups' real-world data. Next slide, please. So over the next couple slides, these showcase what is the current landscape with our key stakeholders, so the regulators, sponsors, and patients, and also what are some of the barriers that they're encountering. So I'm gonna highlight one of the things on these buckets because I know we've all been short on time. So I'll go to the second bullet on to understand the landscape. So CDRH, which is an agency within the FDA, the Center for Device and Radiological Health, they've recently published 90 examples of how real-world evidence has been used within their pipeline of approval. And so they've actually created multiple offices to help handle the real-world evidence understanding. And so they really have become, from my understanding and research, really the leaders within the agency to really determine and develop understanding of how we should use real-world data and leverage it. And they played a major role in helping establish NIST, or NEST, the National Evaluation System for Health Technologies. And I could talk about more about them, but I feel like that's one of the great agencies, or I should say organizations, that's part of a colloquium of different stakeholders that's helped come up with metrics to understand how should we understand real-world data and be able to convert it to real-world evidence. And so there's many barriers that are associated with this. And as our speakers from FDA already listed, it's very difficult to translate real-world data into real-world evidence. And so that's among these barriers that are listed here. Can we go to the next slide, please? So under sponsors, a current landscape. So if you're looking at, there's a test case that NEST produced on their website where Johnson & Johnson used some real-world evidence, and they partnered with healthcare institutions from Mercy, Mayo, and Yale New Haven Health. And so from that work with NEST and with these collaborators with the health centers, with having the data, they were able to, they're set to become the first sponsor to gain an indication expansion solely using real world evidence to, and submitting it through the CDRH agency. And so those numbers there are basically showing the numbers of sponsors approvals based on using real world evidence to gain approval for their indications. And so one of the major barriers for sponsors includes, patient recruitment. So patient recruitment occurs at poor site selection. So from some of the work that I've done with CRS, we've understood a bit more about the clinical trial landscape and how site selection works to establish clinical trial sites. So that's just one of the barriers. And you can think about that now in the digital landscape. So traditionally those physical sites, but now in the digital landscape, it's important to make sure we're partnering with institutions that have real world data of underrepresented groups. So next slide, please. And so with patients, so the current landscape, of course, diversity in clinical trials is poor. There are strong patient advocacy groups that are making a lot of strides and trying to do more health equity work. Patient registries, as we know, is a great resource. And so under barriers, one of the biggest ones is around community trust issues and really trying to understand how do sponsors and regulators build their trustworthiness for these populations. And as I mentioned before, making sure that those health systems that can house that real world data has diverse populations associated. It's going to be important. The next slide, please. So this is the lens that I'm adding to help us pump out these recommendations. So a health equity approach to increase participation. Next slide, please. And so this is just an overview of my company and my partner and how we're focusing health equity to advance for underrepresented minorities across the life care sciences. So next slide, please. And these are just highlighting the partners that we work with my lab at Yale and my alma mater, Howard, and others to actually, and you can see quickly kind of highlighted in yellow what we've done with our partners. And so we can go to the next slide. And so these are the recommendations that I've come up with. And so with regulators, I really think NEST is doing such a great job coming up with the metrics and understanding, how do we change real world data into real world evidence and creating these standards that health systems can start to follow. So the main thing is just making sure that either we can create quotas and provide incentives, breakthrough designations for inclusion of diverse and vulnerable populations that are utilizing that real world data to convert it into real world evidence. And another key one is, FDA seems inaccessible to the general public. And I think that we can take that, pretty soberly because as I've had conversations with my fellows and community research folks, that there needs to be this increased public interaction with underserved communities to show trustworthiness to the public. And as you all know, there are, patient advocacy committees and groups within FDA that allow that, but it's just more about awareness of the current pipelines that exist that allow folks to have that interaction. So next slide, please. So recommendations for sponsors, real world trials is a new opportunity for sponsors to include the data of diverse populations. So implementing health equity best practices is really my top recommendation. And then limiting these benefits. So one of the key ones that I actually found from working with my fellows was this number 2 point here, which is there are these, sponsors could create limited benefits to participants that complete the study, including but not limiting to free products, access to providers for related follow-ups. And so that could be really interesting to make sure African Americans, and as well as other underrepresented groups, feel they're not just left after their data has been used and captured. It's a great way to build trustworthiness and build rapport. So next slide, please. And so then from my work with the Health 360 app and the Morehouse School of Medicine colleagues, they showed that African Americans, the non-medical lay coaches were just as effective as medical ones. So it would be important to create community ambassadors to liaise between the sponsors and subjects to simultaneously build that bridge of communication and trust. And I know I'm running on time. So, and also to provide education about PDUFA and MDUFA and regulatory productions that and patient advocacy committee opportunities. Because I think as the public understands how the FDA works with sponsors and how those regulations help create accountability on both parties, as well as spur innovation, it helps folks feel more process and not just that big pharma is creating something, making lots of money, and FDA is just signing off, because that's not at all what happens. And I think people don't understand that. And so that educating around that could be huge. And also real world experience trainings for minority focused providers and companies that aim to capture real world data and patient generated health data. Because that can then make the process easier to be either incorporated in NEST or another institution like that. So then it can speed that regulatory process and make sure the data of diverse populations is being included in real world trials. Next slide, please. And so I just want to give my acknowledgements. My CERSI mentor is Lindsay Cobbs, who has been amazing. He's in senior leadership at UCB Pharmaceuticals. My PI, Jeffrey Townsend, my business partner, Diana, and a colleague at CRS, Joe, who's been great. And then my two community research fellows through the Care Center, Belanda and Taritha. And then my funding that comes through the Yale Cancer Biology Training Program. And of course, certainly not least, CERSI. I appreciate this opportunity to do this project and explore this work. So thank you all so much.
Thank you, Christopher. That was a great presentation. I'm sure there will be questions in the Q&A part of the presentation. So with that, we'll move on to the last presentation in this session that will be from Stephanie Zawada from Mayo Clinic on blockchain application for feasibility and telehealth for DSCSA compliance. Welcome, Stephanie.
Hi, can everyone hear me?
Yep.
Awesome. So it's such an honor to follow up with Victoria and Christopher and the work that they've been doing. And it's only 10:15 on the West Coast here, and I've already learned a ton. So I'll go ahead and dive right in. And what we're going to be doing is taking kind of a system level look at the data that some of the projects they were actually talking about earlier today. So next slide, please. Thanks. So how did we get here, and what's the background for the rationale for the work that I've been pursuing the past 9 months at Mayo Clinic? So back in 2012, the New England Compounding Center had a huge meningitis outbreak that was caused primarily by a licensure violation. You had one pharmacy in question mass producing prescription steroids rather than filling individual patient prescriptions as they were received. Dozens of patients died of a fungal infection, and hundreds were injured. The majority, one of the larger states that suffered an impact was the state of Michigan, actually, and I made a note there. The mold in the meningitis was very unusual, and it was just an unusual case in general. And so it was easier for regulators and researchers to trace back that source of the outbreak. And next slide. Yes, so that ties back into the state of Michigan, and it was one of the representatives in about September of 2013 who introduced a bill on the House side that had actually a handful of people from his district had died, and it had infected them, and they died of strokes. That was the primary, the main outcome that was associated with that type of an infection. And so Title II of the overall Drug Quality and Security Act was the part that he advocated for. It was bipartisan legislation, was passed and signed into law really quickly, like within a 3-month period. And it set federal requirements for the tracking of prescription medications across the pharmaceutical supply chain. So all the way from like manufacturing to the patient who receives it and disposal there. They proposed a multi-year timeline, 10 years actually, so from 2013 to 2023. It's a full life cycle approach, and they really wanted this interoperable system, which, as was mentioned earlier in the 21st Century CARES Act, the interoperability buzzword has become a real key component to every type of data-driven health work out there right now, but we've yet to see the full realization of an interoperable system in the United States. And it was also aligned to support the Drug Enforcement Agency's initiatives to prevent the distribution of counterfeit medications and respond quickly. And next slide. So there were a broad range of requirements here and there was a great summary I was able to find early on that across the whole life cycle, manufacturers, repackagers, wholesalers, dispensers, and dispensers can sometimes be just a general pharmacy, an independent pharmacy, an in-hospital pharmacy, and some nursing homes can actually count as dispensers. So they have a variety of different requirements to verify the authorization of the group previously that they received it from, so like a wholesaler, or ensure that different trading partners are authorized. It's put lots and lots of different regulations on the different components of the pharmaceutical supply chain. And you also see that they have been forced to kind of adapt during the pandemic. We'll get into that in just a few more slides to try to keep with this 2023 deadline. And next slide. Yeah, so this is just an overview. There have been multiple timeline delays in 2019 and 2020, of course. The FDA adapted some of the timeline to try to give different components of the supply chains, particularly on the manufacturing side, to allow them to continue with their Drug Supply Chain Security Act compliance testing and see if they would have a data system that actually works. And I'm not going to get into the weeds there, but that's a high-level overview of just how complex the process has been so far. And next slide. So there's this serialization requirement. And what is that? So you've got this traceability path that you want to have the actual drug unit, the product in question, whether that's an opioid or a general prescription drug, or even sometimes a generic. You go from the vendor and the wholesaler, the pharmacy, and the patient. And all across that timeline, you want to have metadata that is verifiable and associated with each step of that chain of custody for a prescription. And this serialization requirement specifically mandated a product identifier for each individual unit, each package or each drug case. And that product identifier needed to it must store the following information. It had a couple of mandates there, that it would be a global trade identification number, that there would be a standardized numerical identifier that would have a US national drug code, and then a unique serial number of up to 20 characters. The batch and lot number, so you could quickly determine who else might be affected by either a counterfeit medication or some type of outbreak, and then an expiration date as well. And so that information would stay constant throughout, and some other aspects of it would change as the custody of ownership for a drug unit changes. And next slide. This is where the whole discussion around, well, is blockchain a solution for Drug Supply Chain Security Act compliance? And for those of you who aren't as familiar with blockchain, it's a peer-to-peer public ledger software that's maintained across a distributed computer network. So you have multiple computers with backup information, and it wouldn't just go, if one computer goes down, you don't lose all the information. That's kind of the general gist there. And I like to define a public ledger software as, when you go to a notary public and you get it signed and it's verified, and that record is also stored with another trading partner in a transaction. That's kind of the thought behind, okay, it can't be changed and it's on the record. So it can actually utilize real-time data to track, detect, and respond to harmful prescription drugs. So there have been a variety of cases where they were implemented on small one-to-one little trials, and you have package-level distribution monitoring for a product identifier. That does work, and it works in other industries as well. You have prescription distribution partners that can be all a part of a distributed network. Easy for regulators to review. The data can be shared and shared in a reliable manner. And it also promotes an interoperable electronic system. It's been well received in other parts of the world, and we'll get into a couple use cases and case studies around that. Next slide. So we started with a systematic review to see what exactly was out there and in the United States and then beyond. And we found that there was no research into blockchain-enabled drug supply chain tracking specific to remote prescribing in telehealth, which has become a really big issue, a really big opportunity during the pandemic. And so a couple of the key takeaways were, okay, there's an incremental tracking requirement that it's phased in through 2023 when the Drug Supply Chain Security Act goes into full effect. And research on blockchain as a solution has demonstrated its effectiveness. That was presented just last year in the FDA's Blockchain Interoperability Pilot Project Report on Drug Supply Chain Security Act compliance. So you had one pilot program, which was very well received. It was called Bruin Chain. It was a UCLA ledger domain collaborative work where they actually just pulled commercial off-the-shelf technology, and they found it to be effective at tracking drugs all the way from the manufacturer to the wholesaler and even to the patient and pharmacist. And then there was another just verifiable resource that it was a great kind of review on blockchain technology in the general healthcare sector in the United States. And it outlined examples of companies attempting to record transfer of ownership for prescriptions, and found that you could actually trace, if implemented correctly, blockchain could be a feasible solution. Next slide. So here's an emerging study, and Pharma Ledger, the EU is actually ahead of the United States on this issue, but they also they engage in the exchange of trans-border drug exchanges more frequently than we do here in the United States and Europe. So they implemented a blockchain infrastructure that used multiple chains, like multiple ledgers, that rested on one overall, like interoperable blockchain using the standard blockchain protocol. They built self-sovereign applications, meaning that they weren't just tied to the identity of like a patient identifier. Patients would have control. They were also part of the design, and their individual devices, patient portal access, just the fact that that instance exists, that's a self-sovereign application rather than needing their patient identifier code in a country. They formed this alliance in January of 2020, so just last year, and it was partially funded by the EU's Innovative Medicines Initiative, and it has 29 member organizations, including GlaxoSmithKline, Merck, and Novartis. So they've been piloting and participating in the development of this project in the EU, not in the US yet. So next steps in Europe, actually, it's been successful. They've identified a range of use cases, particularly one that's of interest in the US right now is that Internet of Medical Device trials, just because so many clinical trials were forced to include a distributed component during the pandemic, and we'd like to harness and make sense of that data eventually. So the goal there, moving forward, they've published their next steps, and there was an article in Forbes just a week ago that they want to partner with health authorities across the EU to deploy blockchain-enabled an application that in different pilot markets they had. And the system that they used was built on previous literature, and then a deployment of what they're calling like a pharma ledger that allows patients to download just an app and then scan a little 2D matrix, a code on the outside of a drug package. It's kind of like barcode scanning, but like for an individual person. And it's similar to that QR code, but it stores all that information that's required according to EU guidelines, and it does recognize the manufacturer and then sends a request for the most up-to-date digital leaflet for that particular drug. So it's in early stages, but it has been working, and they're satisfied with the results. And that's in Europe. And if you want to go to the next slide, sorry. So when it comes to preparation on the US side, and we have different regulations, different requirements, but they have similar overlap themes, we have delayed data system testing. And this has been kind of there have been a couple reports published, there have been articles, industry panels, and it's just over the past few months, as right there, that industry panel mentioned just 2 weeks ago, I think, that testing is worrisome. It's delayed on the US side. The US is not specifically mandating that one specific data system is used. Most stakeholders are opting for a global supply. It's called the Electronic Product Code Information Service Standard. And it contains like what, when, where, why event data that updates as that scanning would occur across the delivery of a prescription drug all the way to a patient, so manufacturing to the patient. It would update with that kind of information as it goes. That standard is interoperable, so it works across different systems. And there's a major delay with manufacturers specifically in the supply chain not using it. They're less prepared. The smaller the manufacturer, if it's say not related to GlaxoSmithKline or Novartis, like we were talking about in Europe, they're just less prepared to handle this type of scaled system. And many stakeholders right now have been manually entering transaction data in spreadsheets. Next slide. So just a quick overview. In the US, we have different remote patient monitoring, telemedicine, clinical services, and telehealth. And all of these have had a range of expansion, temporary, permanent, across our entire country. And they vary on a state-by-state and even federal basis. So this is really complicated what the original proposal was, and then we had to scale and adapt our research around just the time changes during the pandemic. If you want to go to the next slide. Thanks. So how did we start to look at the feasibility here? The purpose was twofold. We took the actual compliant blockchain applications and analyzed whether or not it would be a feasible approach, found that yes, it was. Europe was doing this, and we're starting to look at a few testing implementations in the US. And we used the non-adaption, abandonment, scale-up, spread, and sustainability framework for and complexity assessment tool that was actually developed at Oxford. And I was able to partner with them to start evaluating a potential application here in the US. And next slide. Thanks. So I put up in case anybody wanted to go back or ask questions, just kind of the use cases of how that transfer of ownership would occur with a blockchain across the ledger. If you think of just that block in the center as the ledger, and every time somebody scans, whether it's with a phone or a barcode scanner that's specific to the application, you can update and keep non-changing information stored across the entire manufacturer to patient supply chain. And next slide. Going to keep us on track here. So using that NASCAT framework analysis, what were the big barriers that we're facing even just here at Mayo Clinic? So on the technology side, yes, we have that blockchain does work for Drug Supply Chain Security Act compliance. It's a feasible technical solution. Telehealth platforms, though, would need to have these compliant blockchain networks. Like they would have to be enabled to kind of cross that public-private boundary of the different platforms, telehealth services, even remote monitoring tools, apps like that were discussed earlier today, that they would also use if it was something that needed to be tracked. And then again, blockchain scalability is currently limited, so you have to have the built-in capabilities at your organization. Adopters, personal mobile devices on the patient side, and even sometimes on the provider or pharmacist side, they may not be up to scale to scan and continue that supply chain tracking. Organizations, you have, as I mentioned, different consent policies, prescription drug monitoring program issues across state lines in the US. So those have to be like mitigated on a step-by-step basis. The wider system, we have changing interoperability rules, changing reimbursement rules around telehealth, changing EHR rules. It's just a system in flux right now. And then embedding adaption over time. We only have a few states starting to look into how it would actually work, but Virginia and Oregon have taken the lead. Next slide. And next up, so at Mayo Clinic, we have the advanced care at home model. We have our own network deploying that care in Florida and Wisconsin. We were able to outline a handful of small little pilot program projects, use cases there that we would deploy moving forward into the summer and fall of this year. And basically, we were looking at drug delivery sensors that would be able to stream data and track the delivery of a drug actually to patients, and then a mobile app that had barcode scanning features for patients or when they were receive a drug at home, and then also just a general software platform that is compliant and would facilitate the storage of that blockchain data as we go. And next slide. So conclusions, blockchain does work for Drug Supply Chain Security Act compliance. It's currently a little bit limited with the scalability and just industry adoption. There are lots of barriers that we outlined in that framework tool. There are changes going on now that might facilitate its adoption a little bit more, and I think that's something that we've witnessed across just in general in the US with different technologies being more widely adopted. Nursing homes and care at home present unique challenges for that compliance because you're tracking the actual delivery of a prescription to a patient's home, and that's something that we're starting to look at here. And then again, the compliance is costly, and we need more regulatory guidance along these new supply chains that are emerging around remote patient monitoring and digital health and care delivery at home. And so we need to do more research and look forward to doing that in the summer and beyond once we start actually piloting one of the blockchain projects. So thank you very much. And I think the next slide, has thank yous to some of the people that were able to help and collaborate with me over time. Dr. Liu was the mentor there, and I had different rotation mentors in digital health and ethics, and then sponsors at Mayo that helped me also connect with faculty at Oxford to do the feasibility analysis.
That was fascinating, Stephanie. Great presentation. So we have a few minutes now to take any questions or follow-ups from the three presentations in this session. So, either if someone wants to raise their hand or if they want to just put a question in the chat, either way would work to bring up your questions. And maybe while we wait, I'll start out by a specific question. I'll maybe start out with a question for Christopher. Christopher, from, I thought your approaches to sort of increase participation and number of things you laid out was very interesting. The one question I had is how did you think about, as there's increasing focus on virtual clinical trials, as well as there's specific centers where there's more minorities that receive their care, and how to engage them in clinical trials as two different areas of potentially increasing enrollment into clinical trials. And I don't know if you gave some thought to those, especially the virtual clinical trials seem to becoming a bigger and bigger issue. So I just wanted to hear your perspective on both those topics.
Yeah, no, so I'll tackle that first. You're saying establishing sites. So how, like, you're asking recommendations on how to establish sites where or target populations you want to enrich for?
Yeah, so I think that the question is oftentimes clinical trials happen at academic centers or other places that may not have as much representation of African-American patients. And so how do you sort of match up the enrollment sites to sort of increase overall enrollment of more diverse populations?
Yeah, so this is actually something I've worked directly on with my company hat. So we have a client that developed a test, a colorectal cancer test. It's a blood test to detect colorectal cancer. And we actually, well, through our network, we partnered with Howard University, Morehouse School of Medicine, Meharry's College of Medicine, because those are the more premier HBCUs that have medical campuses. And so they're a bit more advanced into actually, and they have clinical trial offices typically, and that's essential to integrating with sponsors to execute the clinical trials. Because to your point, it's very difficult to find providers or academic institutions that may not have, readiness and preparedness to execute on completing a clinical trial. So that was one of the ways we've done it, was partner with institutions that already have some of that infrastructure built, and making sure that that trial could be implementable. Because to your point, what happens a lot is that community centers and academic centers that primarily serve underserved folks are already overwhelmed with COVID in particular that's been going on, whether it's, any other condition that's, been layered. So it's important then for sponsors to streamline their trial design to make sure it can be completed. Because as you know, like, completing enrollment is one thing and then completing the trial is another. So to make sure that then those incentives and that communication is very clear up front is paramount. And then one of the biggest things about the real world data piece is, again, because as you know, there's kind of two ways you can grab it. You can do primary data collection of real world data if you directly involve folks in the beginning and the kind of the pre-market phase, or you can go to the post-market and just go directly to the health centers that have already had their billing and other mobile applications that have their, real world data of those populations. And so those are kind of tricky because you don't want to create a sort of Henrietta Lacks situation where folks are, being like, oh my gosh, I didn't know my data is now used in this application. So there's other things that we have to think about to make sure like that's communicated when a patient enters into a health center to have their real world data captured, how it's going to be utilized, as well as in the primary phase, and they're directly engaging on how that data is going to be captured and what protections do they have. And how will it be utilized and leveraged to benefit not only them, but typically, generations to come. So we've kind of leveraged that, and that's usually been taken on really well by our community partners and folks we've worked with.
Great, thank you so much. That was very, very helpful. And specifically, the institutions you worked with seem like, a very good place to start. And I think those insights then hopefully can be spread to other similar places that may not have as much infrastructure.
Absolutely. Thank you.
Thank you. Any other questions? So while we wait, I'll pose another couple of questions. A question for Vicky. As obviously what you looked at here in the context of wearables and the potential for wearable in wearables and impacting healthcare in the future, especially post-surgical care, could be pretty significant. And so two pieces that I will ask. One is, what is the evidence you think that would be necessary to make that part of routine care? And related question is, as most people have their own wearables, be it the Fitbits, Withings, Apple Watch, et cetera, how do you enable, wearables being agnostic to the underlying wearable to use some of those pieces to impact intervention or early identification of deterioration, whatever that might be? I know they're both difficult questions, but just wanted to hear your perspective as you've been working on this project.
Sure. So I guess the first question about what kind of data is going to be helpful potentially in kind of post-procedural recovery or post-hospital recovery. I mean, certainly there have been studies that have shown like associations between activity and clinical outcomes in like the post-recovery, post-procedure setting, but also not. And so I think like certainly given more studies in specific areas, I think that seems like a promising area. And then, I mean, I guess particularly, so there's like activity, step count. I think that the wearables around like mobile EKGs and heart rhythm are sort of, can be really helpful used in the right context. I think there have been studies that show that, the Apple Watch is like heart rhythm detection function actually leads to not that many kind of new in a context where we know that people have and that's a totally different situation where it can be AFib detected by a wearable is associated with like long-term recurrence. So I'm not really sure how helpful that is, but I think it's important to kind of, with any wearable really, it's important to understand like what are the specific situations where this is helpful and what are the specific situations where it's less helpful. And I think that's something that, is still being kind of worked out. And then, I'm sorry, could you remind me your second question?
So I think the second question was more, as you, here in this context, we were looking at Fitbit data, right? But if you have different wearables and, people are not going to get wearables from the health system. So most people have their own wearables these days. Is how can you have these interventions be a little bit wearable agnostic, given that the underlying algorithms for each of the wearables are somewhat different?
Yeah, it's really hard. And like, not only, are the algorithms for the wearables different, but also the way people use them are different. And you can't really guarantee, a patient is going to put on their wrist rather than their foot or something. And that's the data that you get. And I mean, that's just like, I guess that's inherent in real world data generally. I mean, I know that there have been studies about, the accuracy specifically of different products. And I think that from what I've like seen, even those results are kind of variable and, it's different in different populations. So honestly, I kind of don't really have a great, it is a big question. And kind of the more you dig into it, the more you see that there's variation in the products, in use, in like the patterns of use. But yeah, I think, I don't know, it's a difficult question I'm not sure I have an answer to.
Yeah, no, and I realized that was a difficult question, but I thought, I would pose it in case you or what thoughts you had in general. So I don't know that we have an answer necessarily there. So thank you, Vicky. And last question before we move on to the next session for Stephanie.
Anyway, sorry, just to cut you off. Stephanie actually had to hop off, unfortunately.
Oh, she did. Okay.
Yeah, so sorry about that.
Oh, no problem. Thanks for letting me know, Tiffany. So with that, maybe I will turn it over to Joe to start the next session.
Great. Thanks, Nilay. Some people will do anything to avoid getting in the hot seat with you. I think that's the lesson learned here. I'm going to MC our session 2, and we're going to start the session with a present from Tristan Furnary. I hope I'm saying your last name correctly. Why don't you introduce your project and take it away?
Thank you, Dr. Ross, and a huge thank you to the organizers for putting this event together. My name is Tristan Furnary, and I am a second-year Master of Public Health student in the Yale School of Public Health's Environmental Health Sciences Department. In this presentation, we will be going over transcriptomic and metabolomic analyses of acetaminophen exposure in a stem cell model. And this is all done to explore the potential mechanisms explaining epidemiological associations between prenatal acetaminophen exposure and increased risk of autism spectrum disorder. Next slide, please. Thank you. So according to the CDC, 1 in 54 children born in the US in 2008 have been diagnosed with autism spectrum disorder, or ASD. This is a clear public health issue. And as you can see, the prevalence of autism has been increasing over recent decades. In fact, we can see in the graph to the right from the CDC that the prevalence of autism has almost tripled over the past 16 years. Still, the underlying mechanisms for autism are so far unknown, with a likely complex and multifactorial etiology. Next slide, please. Wonderful, and this is the one with, yeah, thank you for the animations. So recent epidemiological studies using various mother-child cohorts and sibling-controlled cohorts suggest prenatal exposure to acetaminophen, also referred to as APAP, is associated with increased risk of autism spectrum disorder. And if you could go through the rest of the animations now, thank you. Approximately 65% of, this is critically important for public health because acetaminophen is the most commonly used drug during pregnancy. If you wouldn't mind advancing the animations forward a bit more. Thank you. Approximately 65% of women take the analgesic at some point during their pregnancy. Looking at the literature, in vitro studies using cell cultures have shown that concentrations 10 times higher than the therapeutic dose induce cell death. Next slide, please. Thank you. To better understand the potential mechanisms by which prenatal acetaminophen exposure could affect basic processes of human nervous system development and thereby could result in behavioral dysfunction, we used a systems-level approach in human stem cells to investigate the mechanisms by which prenatal exposure to physiologically relevant doses of acetaminophen could increase the risk of autism spectrum disorder. The methods employed in this study therefore fall into category 3D of the FDA Center Regulatory Science Research Priority Areas, as it establishes methods to improve and streamline clinical and post-market evaluation of FDA-regulated products pertaining to their safety and efficacy in perinatal and pediatric individuals, specifically with consideration to neurodevelopment in this case. Next slide, please. So our study initially set out with the aims to conduct this work in both an induced pluripotent stem cell model as well as a neural progenitor cell model, differentiated from those iPSCs, or those induced pluripotent stem cells. While the differentiation process for the neural progenitor cell model was initially successful, unfortunately our cell bank later became contaminated. Therefore, the preceding experiments were conducted only on induced pluripotent stem cells, while neural differentiation troubleshooting was done concurrently. We exposed iPSCs to a range of concentrations from 0.16 millimolar to 1.28 millimolar for 6 days with daily medium changes. And this 6-day exposure period was chosen to simulate a pregnant woman taking acetaminophen for chronic pain. we initially examined the concentration dependence of acetaminophen toxicity in these human iPSCs through a dose response curve as estimated through cell counts, excuse me. So we calculated the LC50, or the concentration at which 50% of cells died, to be 0.64 millimolar. Therefore, we selected the 3 concentrations below the LC50 for further downstream experiments. Those acetaminophen concentrations were identified as the representative therapeutic concentration, which is 0.16 millimolar, and representative toxic concentrations of 0.32 millimolar and 0.48 millimolar. These concentrations, along with a DMSO vehicle control, were used in preceding analyses, which include RNA sequencing and metabolomics on ultra-performance liquid chromatography mass spectrometry, or just LCMS for short. Next slide, please. Thank you. Reads from RNA sequencing were run through a pipeline so that differentially expressed genes and normalized read counts were determined by DESeq2, and then visualized in ClueCore Omics Explorer. Peaks for metabolomics were deconvoluted and identified in MZ-Mine, and then analyzed with MetaboAnalyst. Finally, pathway enrichment analysis was performed on differentially expressed genes and metabolites using Ingenuity pathway analysis. Next slide, please. Thank you. So we were excited to see that several genes that have been found to be dysregulated in individuals with autism spectrum disorder, irrespective of acetaminophen exposure, were found to be dysregulated in the same manner in our results. Furthermore, genes that are downregulated in autism literature are found to be downregulated in our data set. So if we focus here on the left on glypican-4 or GPC4 as an example, what I'm showing is a B-swarm plot indicating the 3 replicates for each treatment, starting with DMSO control in turquoise, 0.16 millimolar in red, 0.32 millimolar in green, and 0.4 millimolar in blue. As you can see, there is a concentration response relationship, whereby if we increase the concentration of acetaminophen, we see a decreased normalized counts for GPC4. For instance, if we look at the 0.48 millimolar concentration, we have an average read count of 13.3 versus the 13.67 average count for the DMSO control. Additionally, we find significant differences between all of the treatment groups as indicated by Benjamini-Horchberg adjusted p-values, which you can see across the top of this figure. Looking at protein 1 or PROS1, we see a gene where there is a clearly concentration response relationship, whereby if we increase the dose of acetaminophen, we observe an increase in the normalized counts for PROS1. Ultimately, RNA-seq analysis revealed acetaminophen concentration-dependent decreases in expression for COBL, CYP2B6, frizzled class receptor 5, GPC4, NCALD, and FGFBP3, and increases in expression for ATP1A2, PROS1, HES1, and NRIP1. All of these changes in gene expression are identical to those described in the autism spectrum disorder literature. And again, that literature is not regarding acetaminophen exposure at all. So these results are unique in that they are almost emulating the autism spectrum disorder literature, but through acetaminophen exposure. Next slide, please. Thank you. Moving on to results for metabolomics, if we look over here to the right, we see an overview of the enrichment analysis. As we move to the right along the x-axis, we have a larger negative log 10 p-value. This is also indicated by the color of the circles that we see, with darker red representing smaller p-values and lighter red representing insignificant p-values. Additionally, the enrichment score for each of these metabolite sets is indicated by the size of the circle. So focusing here on the top 2 metabolite sets, if you wouldn't mind adding the animation. Thank you. Metabolomic analysis revealed acetaminophen exposure to lead to differential regulation of metabolites involved in aminoacyl tRNA biosynthesis, as well as valine, leucine, and isoleucine biosynthesis, both of which have been linked to autism spectrum disorder. Next slide, please. Thank you. Analyzing the differentially expressed genes and metabolites concurrently via IPA revealed two significantly overrepresented canonical pathways of interest. The first was nuclear factor erythroid 2 related factor 2 mediated oxidative stress and serotonin receptor signaling, both of which have been shown to have direct implications to autism spectrum disorder. What's really exciting is when we use IPA to combine and build on top of our RNA-seq and metabolomics data set, we can see that there are certain functions where they are predicted to be inhibited, as shown by the blue octagons, such as morphogenesis of neurons, branching of neurites, dendritic growth or branching, and quantity of neurons. Alternatively, this tool also allows us to predict activation of certain processes, which appear as the orange octagons in this figure, and include proliferation of neural cells, growth of neurites, and growth of axons. The three diseases of interest identified through this pathway analysis and represented as the white crosses in this diagram are autism spectrum disorder or intellectual disability, cognitive impairment, and glucose metabolism disorder, the latter of which has been implicated in the etiology of autism spectrum disorder. So it should be noted that each connection in this diagram represents a significant relationship supported by findings in published literature, while the yellow connections actually indicate a finding that is contrary to what would normally be expected based off of that literature. Next slide, please. Ultimately, our integration of transcriptomics and metabolomics data has revealed an impact of acetaminophen on biological processes that appear to parallel those seen in autism spectrum disorder. In doing so, the results of the present study provide an important first step in highlighting the potential mechanisms by which prenatal exposure to acetaminophen may increase the risk of autism spectrum disorder and promote adverse neurodevelopment outcomes in general. Some limitations of the present study include the low power of this research due to the chosen low acetaminophen concentrations. However, these concentrations are most representative of real-life exposures and therefore justify their use in the present study. And that essentially is the whole motivation for exploring this work with the lower concentrations to understand the effect of acetaminophen exposure at what is physiologically relevant. Another limitation is the fact that an in vitro iPSC model is being used to infer the effects of prenatal acetaminophen exposure at the in vivo level. However, we feel that through the continued development of the NPC model, we will be able to overcome this limitation to the best of our ability, in that we are going to design a model that is more specific or specialized for neural development. Next slide, please. So the results presented here suggest how prenatal acetaminophen exposure may relate to published literature on autism spectrum disorder, and may also elucidate areas or new perspectives for future autism research. The multi-omics methods used in this study, combined with the NPC model currently under development, can serve as an effective tool by which other chemicals of neurodevelopmental concern may be assessed. Future research and clinical implications stemming from the methods employed in this study could also include developing iPSC lines for individual patients from their biological samples, and then that could be used for precision medicine, for example. Next slide, please. Finally, I would like to thank my lab for their support and mentorship throughout this long research process, especially Dr. Vasilis Vasiliou and Dr. Abha Gupta. And of course, I would like to thank the Yale-Mayo Clinic CERSI Scholars Program for their generous funding, without which this research would not be possible. Thank you.
Thanks, Tristan. Again, just to remind everyone listening in, please put questions in the chat. We'll do questions at the end of the sessions. The next presenter is Paul Jacobs. Paul, please introduce yourself and your project.
Thank you. So my name is Paul Jacobs. I work with Dr. Andrew Fagan in the Department of Radiology here at the Mayo Clinic. And my project is an investigation of SAR values induced near an orthopedic implant at 7 Tesla relative to lower field strengths. So to give a little bit of background on this topic, 7 Tesla MR imaging of patients with metallic implants still remains a major area of concern, primarily due to lack of device testing. And this is due to 2 primary factors being magnetic field forces on the implants, as well as RF-induced tissue heating as a result of the implants. A little bit more in depth on that, a large portion of the patient population have these types of implants, which can impede the clinical adoption of ultra-high field imaging at 7 Tesla. There's also an assumption with this that the higher field strengths will lead to higher RF-induced tissue heating adjacent to these implants. Next slide, please. With that said, there have been previous studies that have looked at this, including simulated and physical phantom measurements by fiber optic temperature measurement or MR thermometry experiments. However, a lot of these studies used very complex geometries for their implants, such as the examples in the images you see below. They lacked a focus on controlling for their E-field properties in terms of magnitude and phase along the dimensions or length of their implants. Also only focused on one particular field strength. So they either looked at the low side or at 3 Tesla, or they looked at the high side at 7 Tesla, but rarely both. Next slide, please. So with all that said, the aim of our experiment here was we wanted to be able to investigate the maximum local induced specific absorption rate, or SAR. And when I say SAR, it's really just the amount of energy going into the tissue volume itself. We wanted to be able to look at the SAR rate near an orthopedic implant as a function of both magnetic field strength and the implant length. And to do this, we wanted to be able to model a simulated system which created a constant background E field along the length of all of our implants tested. Next slide. So to give a little bit of detail about the methods we used in terms of our actual simulations, our electromagnetic simulations were performed using a finite difference time domain solver known as Sim4Life. Within the simulation environment, we created dipole antennas at each field strength that were used to ensure a homogeneous as possible E field in terms of the magnitude and phase of that field. We also went through and varied our field strength from 3 Tesla to 7 Tesla by adjusting the actual dimensions of the dipole antenna itself. We also went through and took our orthopedic screw length and varied it from 18 millimeters to 183 millimeters over the 24 distinct measurements. We also decided to simulate in a volume of tissue set to the dielectric properties of bone, primarily because these types of implants, these types of screws would normally be placed in a relatively high bone content environment. Next slide, please. Giving a little bit more detail about the simulation execution itself. Once we had our models set up, we went through our gridding and voxelization process, where gridding is primarily just determining how high of resolution to actually set our model to. And then the voxelization process is really just kind of discretizing the area into cubes that the solver will actually solve the equations across. Once we've gone through and actually run the simulations, we were able to pull our E-field data and our SAR data seen in examples to the right, and you'll see examples on the next slide. So these are some example data. This is some example data that we pulled from our actual simulations. And on the left, you can see that in our background simulations that we ran, background meaning without any implants, we wanted to be able to make sure that our E-fields were tangential along the length of where the implant would be, denoted by the red box, as well as they were relatively constant in magnitude. So we were able to confirm that with our background simulations. On the right is an example of an E-field distribution around an example implant. And then below that is the resulting SAR distribution, where you can see it denoted by the white arrow that the maximum E-field, or the maximum SAR, is typically found at the either end, primarily actually at the tip of the implant. Next slide, please. So before we actually present the results, I needed to go through and do a post-processing analysis on our results to make sure that each one was framed in the correct context. So to give a little bit of context about what I mean by this type of normalization, MR scanners will usually increase their power level until a certain flip angle or a certain reference signal is achieved. In this context, what we did was we increased the power level of our own dipole antennas until a constant B1 plus magnitude, or that by analogy, that flip angle was achieved in the absence of the implant itself. After these power levels were determined, the input powers were applied to each of our simulations.
Oh, sorry. For our patients and staff.
I'm so sorry about that.
For everyone's safety, we require the patients.
At least we know where you are.
Right. I'm so sorry about that.
No.
So after we determined our input powers, we applied those input powers to each dipole antenna specifically to determine the 1 gram SAR that was measured typically at the tip of the screws. One thing to note is this graph in the bottom center of the slide here, which shows that the actual E field magnitude changed, was not as constant as we would have hoped it would have been for each along the length of where our implants would have been. This was something we wanted to be able to confirm, but it didn't really play a huge role in our actual results. Next slide, please. So to actually dive into our results a bit more, after we performed our B1 plus normalization, we were able to actually go through and look at where we were seeing the maximum SAR rates at what specific field strengths. So we can see here is that our 4 Tesla had actually the highest peak. Next slide, please. With our 7 Tesla actually having a 36% reduction in terms of the 1 gram SAR that was measured. Next slide, please. With the after this, the 5 and 6 Tesla had similar but slightly higher maximum SAR compared to the 7 Tesla. Next slide. And then the 3 Tesla actually had the second highest peak compared to the 4 Tesla. Next slide. So what this shows is that the 4 Tesla and 3 Tesla had the higher peak SAR values relative to the 7 Tesla, or the highest field strength that we actually used. And something else to note about the data that you see here is that this data is in the form of these kind of these curves or kind of these humps, and that's actually a resonant effect that we actually expected to see. And when we look at that a little bit closer, we were able to see that those resonant effects in relation to what implant lengths they occur at happened at approximately 1/3 of the operational wavelength of these dipole antennas. Next slide, please. We actually decided to take our analysis one step further, and we wanted to be able to perform a normalized SAR elevation. So what is the significance of this normalized elevation? It mainly doesn't just look at the actual SAR of the implant itself, but it also looks at the background SAR. So that slide I showed you earlier when we just did background simulations, we wanted to be able to take that into account as well in our data. So what we can see here is that when we perform that for each of them, the 3 Tesla actually has the highest SAR elevation. Next slide, please. With the 4 Tesla having actually the second highest. Next slide. The 5 Tesla having the 3rd highest, and so on and so forth for 6 and 7. And this kind of just shows a very linearly increasing, a very nice trend where we can see a linearly increasing SAR value as we go from 7 Tesla and decrease the field value actually up to 3 Tesla. So that's very interesting. Next slide. We also decided to do one final batch of simulations where we wanted to be able to see how the tissue dielectric properties, primarily the conductivity and permittivity of these tissue, determine the resonant peak location and amplitude. So we performed this final batch of simulations all at the 7 Tesla field strength, so no other lower field strengths either. And what we found was that adjusting the conductivity value of these tissues can actually shift your resonant peak it can shift it from a lower implant length to a higher implant length. And then by looking at the actual permittivity values, we were able to see that it actually adjusts where the magnitude of that peak. Where actually, if you have a lower permittivity value, you'll end up having an actual higher SAR peak. Next slide, please. So to kind of go into some discussion about the results that we're seeing here. In general, the RF power required to achieve this constant B1 plus value that I've discussed here has 2 primary competing mechanisms that are working with it. It's been known commonly that the RF power, the radio frequency power of your system, will typically increase, or the amount that is needed will increase as your main magnetic field or your B0 field increases. However, because our dipole length in general is changing size, particularly decreasing in size as our main magnetic field increases, the energy density per unit of power is also changing. So that's kind of why we saw after we did our normalization, the 3 Tesla required more power than the 7 Tesla, this is a possible explanation for that. We also saw, when I showed you the graph of the E-field magnitude along the length of the implant, there was a larger variation of that E-field magnitude for the 3 Tesla dipole relative to the 7 Tesla dipole. And this differential increase in peak SAR between 3 Tesla and 7 Tesla may have been larger had that been taken into account. One possible explanation that we have for why we were seeing the phenomenon that we're seeing in our results is as you go from a lower field to a higher field strength, you're going to go to a higher RF frequency. And because of that, the surrounding medium will increasingly dampen the currents or the induced currents in the surface of the implant, thereby lowering the scattered E field at the tip of the implant and hence lowering the peak SAR at that point. This has been seen in a previous study where the peak SAR was actually seen between 3 Tesla and 7 Tesla, particularly at 4 Tesla. But this study was highly dependent on the coil type, the load, the sample geometry, as well as the number of transmit elements. So it's not exactly one for one. So kind of good to go into conclusion and kind of sum up the results here. We aimed to have a model system to investigate the tissue heating controlling for tangential E-field magnitude and phase along the length of all of our implants simulated. What we saw is an expected resonant SAR behavior observed as a function of the implant length itself. And we also saw that the peak 1 gram SAR was lower at 7 Tesla compared to lower field strengths when normalized for background B1 plus magnitude. The primary output of this study, however, is that while this study was done under very specific experimental conditions, but it does show that there is a need to revisit this assumption that heating is necessarily worse at higher fields for implants. And so with that said, I'd like to give a couple of acknowledgments. I'd like to thank Eric Borsch, who's a colleague of ours who really helped in some of the during some of the technical difficulties we had with the simulations. I'd like to thank the CERSI program for allowing me to present today, as well as the Mayo Clinic Prep program for supporting this project. Thank you.
Thanks, Paul. That was terrific. And we have one more presentation in this session from David Jiang. David, you want to introduce yourself and present your project? You're still on mute.
There you go. Thank you, Dr. Ross. My name is David Jiang, and I'm an associate translational informatics analyst at Mayo Clinic. And so I primarily work with Dr. Shah and Dr. Rosalina McCoy, and I'd like to thank everyone here for the opportunity to share our project today on enhancing FDA regulation of artificial intelligence and machine learning algorithms. And next slide, please. So next slide, please. Before we get started, I'd like to acknowledge the Yale-Mayo CERSI program for funding, James Hahn for his assistance in coding, Lindsay for her administrative help, along with Mr. Morton and Doctors Anasia, Xiang, Noseworthy, and Ross for their guidance and input on this project, and Dr. Shah for his mentorship. Next slide. And so, as you may know, artificial intelligence and machine learning are increasingly being applied to new and novel areas of medicine. However, the evidence of their clinical and therapeutic benefits that these devices bring is weak. And so these applications, as they continue to grow, it becomes more important for the FDA to regulate them for their effectiveness and safety. A little bit of background. Traditionally, the FDA has categorized medical devices into classes according to their potential risk for the patient involved. And class 1 are devices with minimal risk, such as Band-Aids. Class 2 are those that involve some risk that need to be proven to be safe and effective, such as CT or MRI scans. And then class 3 are those that can involve serious risk of injury or death, such as implants. And as such, the regulatory approval process for each class is different. Class 1 devices generally don't require much regulation. Class 2 devices require the device manufacturer to demonstrate what's known as substantial equivalence through a 510(k) clearance to a predicate device, meaning the new device have to perform better or equal to a device that has already been approved by the FDA. And class 3 devices need to go through pre-market approval, where it requires rigorous proof that the device is safe and effective. The last method, called de novo pathway, is for devices that are novel and do not fit into any category. And so in this case, the FDA assesses the risk levels, and then they are approved through the appropriate approval framework, excuse me. And so when a new AI ML device comes onto the market, it is generally categorized. I'm still in the previous slide, by the way, sorry. When a new AI ML device comes onto the market, it is generally categorized as a class 2 or class 3. So for those devices that are categorized as class 2, the manufacturer will then submit a 510K clearance. However, when a AI ML device is categorized as class 3, or if the manufacturer cannot identify a suitable predicate device, the manufacturer will most likely file a de novo classification. And de novo requests allow the manufacturer to argue that the device in question does not pose as much risk as other devices in class 3, and then therefore should be reclassified along with the 2 other classes. And so the existence of the de novo reclassification process means that a vast majority, if not all, of these devices are approved through class 2 and do not go through. Therefore, it is important to know what are these standards that are used to approve these devices in the first place, and which is what this project focuses on. And one of the biggest hurdles to study topics related to this is that there's not a standard data set of known AI devices that the FDA has already approved. And so one of the key components and to study this project properly is that we needed to create our own. Next slide, please. So there have been 2 previous studies focused on this topic. In 2020, Benjamins and colleagues identified 29 AI devices through searching the FDA database for relevant terms, and another 35 through website announcements and press releases. They categorized them into medical specialties, but there weren't much discussion on the data or study type that was used to approve them. And earlier this year, Wu and colleagues looked at 130 devices and briefly looked at whether the study was retrospective or prospective. However, it stops short of deciphering the standards used to approve these devices. Next slide, please. And so the project today has 2 aims. First, we seek to build a complete data set of FDA-approved AI/ML devices, after which we will try to analyze and evaluate the measures of testing and the data used to approve these devices. Next slide, please. So to construct the data set, we first will download all of the published FDA summaries of devices approved through the 510K, de novo, and pre-market approval pathways. Then we will search through the summaries for key terms, excuse me, that regulate AI/ML technology. Then we will manually read through and narrow these down to our final data set. Next slide, please. Excuse me. To abstract the data, we read through the summaries of the devices in our data set and looked for key information. And most of these, the most important to note here were the characteristics of the study. So whether or not they reported the study size, what the results were from clinical testing, if there were any clinical testing, and what type of data was used to support that. Next slide, please. So for results, from over 80,000 initial device summaries, we were able to, through the key terms and sort of an automatic filter, narrow it down to 145. And then we cross-checked our list with the ones that Benjamin and Wu had found. And we arrived at a final list of 182 FDA summaries. Next slide, please. So of these 182 summaries constituted about 135 unique devices from 104 unique manufacturers. And most of these are from the United States, Israel, and the United Kingdom. For one thing to keep in mind is that the vast majority, that's 173 of these devices, were approved through the 510K clearance, and this was expected. 9 were approved through de novo, and we found some, and then we virtually found none that was through the pre-market approval process. The reason why I have an asterisk there on the 0 is that Benjamin's said that they had found 1 device through PMA, but after reviewing that device's summary, I don't think it technically qualifies as a artificial intelligence. It did not fit any of our inclusion criteria. And I am aware of some devices that are in development, such as Medtronic's artificial pancreas and personalized, what they call personalized closed loop insulin systems that recently received the breakthrough device designation, but that hasn't had a chance to go through formal approvals. Next slide, please. And so as expected, a vast majority of these devices are in cardiovascular medicine and radiology. I have not had the chance to go through and give a more granular analysis of the radiology component. We're working on that, but we expect that to be broken down to MRI, CT, or X-ray. And so next slide, please. Out of the 182 devices, only 88, or about 48%, presented any clinical testing data, and the rest only presented non-clinical. And within these non-clinical data sets, most of these devices, even if they presented clinical data, included some form of performance testing. That is, they tested the device to make sure it works through validation and verification, and some bench testing to make sure that the device is safe. About 45% of the devices had risk analysis, where the device manufacturer laid out the risk of the patient's privacy information and other ways to mitigate such risks, and about half included voluntary industry or FDA standards that they adhere to while developing the device. And only one of these summaries indicated that they had established what's known as a change protocol for the AI device. Next slide, please. So in terms of clinical testing, the vast majority that had clinical testing, so that's 92%, were retrospective. The other 7 were prospective. 6 devices had a known registered clinical trial number, and 39 devices indicated that they had used multiple sites to conduct their clinical analysis, with the median number of sites being 3.5. And then 83 devices, their summaries reported their study size, with the median being 286. This 286 either subjects, so people, or 286 physical MRI scans or CT scans. The specific endpoints or evaluation parameters used by these devices were mostly in the form of sensitivity and specificity. Some discussed their devices' time to notification or time to open, and still fewer conducted human factor testing, such as ease of use or training. Next slide, please. And so our study makes clear that most of the devices are approved through 510(k) clearance, whereby the standard of proof only needs to be that they need to perform as good as or better than another previously approved device. These devices generally do not report clinical testing data. Only 7 were prospective studies. There was one instance of a device reporting a change protocol in which they specified how they would guard against unwanted or unplanned change in the algorithm. And despite the clinical studies being reported, most of these results only specified sensitivity and specificity, which was then used to compare to a predicate device. And so these kind of clinical testing data do not really specify any clinically relevant outcomes. And so additionally, the nature of the AI is not reported anywhere within the summaries. So they mostly state something along the lines of, we utilize a neural network, or we used a machine learning algorithm, and that is very vague. At least in my opinion, that is very vague. So next slide, please. So while the results from the study are very preliminary, we can extrapolate several policy recommendations that could be implemented. First, for better regulatory and consumer transparency, we recommend that the device that use AI/ML should clearly state that they do on their applications, and the FDA should keep a list of such devices available. We also recommend for these devices to go through prospective studies and test for results that are clinically relevant, and have outcomes and endpoints that are clinically relevant, and not just comparisons to existing devices. Lastly, we recommend manufacturers to develop a approved change protocol where they state and disclose how they expect the device is going to change, especially in the future when we expect what's called unlocked algorithms, where the algorithm itself actually learns and changes as it gains new information. Next slide, please. So because of how most of the devices are approved by comparison to a predicate device, we don't really know what the clinical outcomes, as we said before, of these devices are. And the next step is to dig more into what are these characteristics of predicates if they're so important in the approval process. So I just took 5 random AI devices, they're in blue. And as you can see, I traced them back through the predicate web, and they're all linked. And a surprising number of them are interconnected in this web of predicates. So there may be some fascinating information to be learned just by analyzing this web across the 182 devices that we have identified. Next slide, please. And so in conclusion, we constructed a data set of FDA-approved AI/ML devices that gave us a preliminary look into the standards of assessment, testing, and approval of these devices. And further, we recommend specific policy changes and outline a number of courses for future study. So thank you so much for your time, and happy to take questions.
Thank you, David. Oh, what a tangled web that's being woven. I guess you could call that paper when you get to it. So
that's a good idea.
Hope maybe the 3 speakers could come on camera. Are there questions from anyone on the seminar? David, maybe I'll start with you just as a step. Obviously the growth of AI is tremendous and we're seeing more and more applications. There is always this sort of challenge of going back to the older and older predicates and its relevance to today's clinical practice. What would be the best way? I mean, you can't do a clinical trial for every one of these. So, is there a sort of a relatively feasible compromise that doesn't inhibit patient care, allows for innovation? I mean, I'm sure that our FDA colleagues would say that is the challenge for them.
Mm-hmm. Yeah, so in the process of tracing back these devices, I went back to like 1983, right? And most of these devices don't even have summaries anymore. So I don't understand how specifically they looked at these devices. And most of how they did compare was they just ran 2 sets of data, the same set of data, and across 2 different devices, and say they perform similarly. And while that is one way to do it, I just don't see how, again, as you said, if that's clinically relevant to anything that we can, we're 40 years now past when some of these devices were first put on the market. So one way to do it is say, you have to use a device within the last, 5 years that's been developed. That's one way. Another way to go about it is to say, well, if, we need to change the definition of like de novo to make sure that the definition of new, right? What is considered new and novel, and make that a little bit more restrictive so that, to make it a little bit more restrictive in that people just don't go the easy way and try to find a clinically, what they would call substantially relevant predicate, make that process a little bit harder to convince more people to go into de novo and to go through a bit more rigorous sort of process for regulation. So that's how I would think about that problem.
That's great. Tristan, maybe you could talk about, probably many of us on this call don't do basic science or haven't since we were very junior in our career. The idea of extrapolating from in vivo or in vitro data to see, that's how inexperienced I am. I'm not sure which it is. It's obviously a challenge. Can you talk us a little bit through the use of negative control endpoints as you're sort of trying to reinforce, or how you sort of triangulate it? I think there are probably some similar lessons to those of us who do observational research with large healthcare data, and we're looking for similar patterns. I'd just like to hear your thought process a bit.
Of course, yes. So I guess really the main negative control we had was the DMSO vehicle control that we used. And the reason for this was the acetaminophen needed to be dissolved in DMSO for it to be delivered at the highest concentration, or a stock concentration could be created, and then from that we could create dilutions. But ultimately that DMSO percentage, volume per volume of the DMSO in the stem cell media was 0.05%. So we feel very confident that that negative control didn't have any major implications for our readouts. And so we felt confident in comparing the acetaminophen exposure groups to that DMSO control. But in terms of, like you mentioned earlier, going from that in vitro to in vivo, that really is the main challenge here, especially because, pregnant women are considered a population in which you would not normally conduct a clinical trial with. So for that reason, I feel that the stem cell technology and methodologies that we can use to emulate that relationship between a pregnant woman and the developing fetus as best as we can are likely it's almost like our best bet in moving forward to emulate that system and understand developmental endpoints and certain exposures.
Yeah, and I can tell you in the pharmacoepi world, that's such a difficult exposure to study, right? Not only is Tylenol can use it, but also, it's preferentially used because we avoid NSAIDs in pregnant women for lots of other reasons. And so there's this sort of risk benefit. Paul, to try to talk about Tristan's project is one challenge for me, but talking about yours is quite another. Maybe you could just talk a little bit about the sort of real world implications of your work, and maybe when FDA should expect to see a PMA using your technology.
Right. So the main advantage behind this type of the simulations we did, right, as I mentioned, were under very specific conditions, where basically we were looking at one particular box of tissue with an implant in it and kind of characterizing how does the SAR level around that implant change as you change the field strength or the implant's one particular dimension of the implant size. But this type of method can actually be used a little bit more advanced, where it's becoming more common to see virtual human body models. So basically, there are whole companies out there, like commercial companies, that have gone to the effort of actually taking, for example, CT scans of patients, segmenting them out, and building these virtual body models where all the different tissue can be is accurate to their electrical properties, and you can actually stick an implant in, for example, like the wrist, and you'd be able to see what kind of SAR you would expect while having the complicated tissue structure that would be around the implant itself. So that's kind of where I see it going more and more, because doing physical patient studies, it's a little impractical when you're trying to determine what kind of implant can be used without harming the patient. So I do think that that's probably the next step that a lot of these manufacturers could take to be able to get their devices at least MR conditional for ultra high field, particularly 7 Tesla and above.
That's great. Thank you. And I saw there's some questions back and forth between David and one of our attendees. But I think I want to just thank all three of you for presenting your work today. It's really terrific. That shows the breadth of work that's been done across our institutions that related to regulatory science. So thank you. And I'll hand it back to Nile for the third session.
Great, thanks, Joe. Those were 3 really great presentations, and now this is our last session of the day for 3 more presentations, and we're excited to start with the first presentation from Montserrat Lara-Velazquesz on SERPINA3, a key player in glioblastoma malignancy and a potential biomarker for the disease. Montserrat?
Thank you, Dr. Shah. I'm an MD PhD graduated from Dr. Guerrero Casares Laboratory in Mayo Clinic, Florida. Our project is entitled SERPINA3, a key player in glioblastoma malignancy and a potential biomarker for the disease. Thank you. The overview of that slide, thank you. The overview of my presentation, I'm gonna talk about a brief introduction on glioblastoma, some facts and statistics. I will also be talking about the relation between periventricular glioblastomas and the relation with the subventricular zone. Posteriorly, I will be talking about the effects of the cerebrospinal fluid on proliferation and migration of cancer cells. I'll be showing also the results of a microarray experiment, how the cerebrospinal fluid can actually regulate the transcriptomic of cancer cells. And I'll be showing the results of a proteomic analysis, the protein composition of cancer and non-cancer cerebrospinal fluid samples. And at the end, I will be talking specifically about our target, which is SERPINA3, the clinical implications of this molecule, as well as how it can be regulated by the cerebrospinal fluid and the in vitro effects and in vivo effects of SERPINA3 in migration, invasion, and proliferation of cancer cells. The next slide, please. Can you advance the animations, please? Yeah. Thank you. So glioblastoma is the most common and aggressive form of primary brain cancer in adults. It accounts for 54% of new gliomas and 45% of primary malignant tumors. In the States, it has an incidence of 2 to 3 cases per 100,000 inhabitants. It's slightly more common in men than women, and nowadays the treatment for glioblastoma includes surgery, chemo, and radiotherapy. Also, the FDA just approved tumor treating fields, but it's not widely used yet. However, despite these aggressive treatments, the survival expectancy of GBM patients, it's only 14 months. And the recurrence rate is close to 100%. Next slide, please. Besides histological characteristics of glioblastoma that contributes to its malignancy, also tumor location is an important factor for the disease. It has been shown by several groups that tumors that are in closer contact with the lateral ventricles exhibit worse survival, and they tend to recur in distant sites to the primary tumor. Next slide, please. Thank you. The main reason for these periventricular tumors to be more aggressive is not known. However, several hypotheses suggest that maybe the interaction between these tumors that are closer to the lateral ventricles with one of the main neurogenic niches in adults, which is located in the wall of the lateral ventricle, which is called the subventricular zone, may be a contributing factor for the aggressiveness of periventricular glioblastomas. Next slide, please. Thank you. So I'm going to be showing the results of our project on what are the factors involved in this in increasing this malignancy of periventricular GBMs. The next slide, please. With those facts in our mind, we starting elucidating what was the role of the cerebrospinal fluid in cancer cells. Our first approach was to analyze how the cancer and non-cancer cerebrospinal fluid was affecting the migration and proliferation of cancer cells. What we did here with the Boyden chamber experiment, we saw that cerebrospinal fluid derived from GBM patients actually increased the migration and proliferation rate of cancer cells when compared with the control condition, which is only media, or with cerebrospinal fluid derived from patients without cancer. Next slide, please. Thank you. We then also wanted to see if the cerebrospinal fluid had in fact a role in modulating the transcriptomic changes of cancer cells. What we did was to condition during 24 hours cancer cells with cancer cerebrospinal fluid, and as you can see here, we got several up and down regulated genes. Interestingly, one of the genes that were upregulated have been already described and had been linked with cancer, such as ALDO-C, CD44, and osteopontin. And as you can see here, we also got SERPINA3, which was among all the genes very upregulated in our cancer cells in response to the cancer cerebrospinal fluid. Next slide, please. Thank you. We also wanted to see what was itself the proteomic composition of the cerebrospinal fluid samples. And for this, we collected high-grade, low-grade, and non-cancer cerebrospinal fluid from different patients with the help of the neurosurgery department at Mayo Clinic. Next slide, please. And while we analyzed the data, we saw that in one of the main cluster of proteins that was shared in common among the cancer cerebrospinal fluid samples, we again got this target molecule, which is SERPINA3. Next slide, please. Thank you. So what is this SERPINA3, or also known as alpha-1 antechemotrypsin? It's a serine protease inhibitor that inhibits mainly cathepsin G and mast cell chemasas. It's synthesized actually by different cells in the body, including hepatic cells, astrocytes, and endometrial cells. And interestingly, it has it's increasing in inflammatory processes, neurodegenerative diseases, and cancer. Specifically talking about cancer, SERPINA3 is overexpressed in endometrial, melanoma, gastric cancer, and in glioma. Next slide, please. Thank you. And while looking into the literature, we saw that in glioma specifically, this expression of SERPIN has been linked with grade. Higher the grade of the glioma, higher the expression of the protein. Next slide, please. Thank you. So our initial approach was to confirm by looking into the TCGA database available online if the expression levels of SERPINA3 were actually upregulated in comparison with the non-cancer tissue. And as you can see here in the first graph to the left, upper left, we saw that the GBM tissue actually had higher mRNA levels than the non-cancer samples. And in the graphic in the middle, we saw that in fact, the expression of this protein was related with glioma grade. In the upper right graph, you can see also that we saw that GBMs with higher expression of SERPINA3 exhibit lower survival. And with the help of the neurosurgery department, we were able to confirm in our primary brain tumor samples that SERPINA3 was upregulated, again, at the mRNA level, and we also confirmed that through at the protein level by this Western blot, as you can see here in the middle. Thank you. Then we wanted to see if the cerebrospinal fluid had in fact also a role by promoting the expression of SERPINA3. What we did was to condition cancer cells with this cancer cerebrospinal fluid, and in fact we saw that this conditioning overexpressed the mRNA levels of SERPINA3 in 4 different primary brain or glioblastoma cell lines. Next slide, please. Thank you. In order to understand more the biological implications of SERPINA3 in glioblastoma, we perform knockdown or silencing experiments as well as overexpression experiments. In this first set of graphs, what we did was to knock down or silence the expression of SERPINA3 in cancer cell lines, and then we evaluate the migratory and proliferative capacity of the cells. As you can see here in the first graph on the left, upper left, while after we knocking down SERPINA3, the cancer cells are not able to migrate as much as the control. And then we also decrease the same capacity, but for invasion. They are not able to degrade extracellular matrix to move from one place to another. Yeah, that slide, thank you. We also saw that by knocking down SERPINA3, we were able to decrease stem cell characteristics, including tumor sphere formation, as well as proliferation by inhibiting the cell cycle in cancer cells. Next slide, please. Thank you. So we also overexpress SERPINA3, and for this you have to take in mind that SERPINA3 is already overexpressed in cancer cells, so we even increase even more the expression of this protein in cancer cells, and we saw that this actually affected positively the migratory capacity of cancer cells. We made the cancer cells more aggressive by increasing the expression of SERPINA3, and they were able also to invade faster, meaning degrade extracellular matrix and move faster while by overexpressing SERPINA3. Next slide, please. Thank you. And then we wanted to check if extracellular SERPIN used as a recombinant protein had any role also in modulating the behavior of cancer cells. And we saw that the recombinant SERPINA3 was promoting also the migration in cancer cells, and this was tested in 3 different primary glioblastoma cell lines. Next slide, please. Thank you. So now, once we confirm in vitro that SERPINA3 had a important role in glioblastoma malignancy, we move forward with in vivo experiments in immunocompromised mice. Next slide, please. And in here, we just confirmed what we saw in vitro. We implanted in immunocompromised mice cells that were silent for SERPINA3, that were not downregulated for the expression of SERPINA3, and we saw that these mouse were able to survive longer than the controls as well as we saw a decreased proliferation index in these mice with tumors where SERPINA3 was knocked down. So this is really telling us that more than the in vitro results, it has actually an in vivo role in affecting survival, this protein of interest. Next slide, please. So with all these conclusions, we think that if this SERPIN actually can be used as a biomarker for GBM, which is important stuff to be, and is the translational point of view of our project. Next slide, please. So with this, we started first by checking the levels of SERPINA3 in non-cancer, in cerebrospinal fluid from non-cancer patients, patients with low-grade gliomas and patients with high-grade gliomas. And interestingly, we saw that the patients with high-grade gliomas have higher expression of SERPINA3 than the non-cancer patients or the patients with low-grade gliomas. This, again, is a starting project. What we are gonna do next is to evaluate in matching samples the levels of SERPINA3 both in plasma to see if there is actually any correlation in the levels of SERPINA3 between CSF and plasma. Next slide, please. And then this is still ongoing. We are actually doing volumetric analysis of brain tumors according to a classification that was published by Lim and colleagues in 2007. We want to see if tumors that are in closer contact with the subventricular zone with the lateral SERPINA3, we want to evaluate if there is any correlation with these periventricular tumors and if SERPINA3 has a contributing role in the malignancy of these tumors. Next slide, please. And finally, in these tumors, we want to evaluate also the genetic mutations with expression levels of SERPINA3. This is information that we collected from the TCGA databases and GlioVis, and as you can see here, the expression levels of SERPINA3 tend to have a correlation with the presence of genetic mutations that are actually really important for tumor malignancy in regards of response to treatment and therapy resistance. Genes such as EGFR, PDGFA, MGMT, IDH1, and TP53 have been really linked with the worse survival in GBM patients, and some of these are overexpressed even in glioblastoma patients. So we actually believe, in fact, that SERPINA3 might have a role also in the presence with these genetic mutations in glioblastoma patients. Next slide, please. As a general summary of the project, we can conclude that there are pro-migratory effects of cancer cerebrospinal fluid on GBM cells. We detected higher presence of SERPINA3 in GBM samples at the mRNA and at the protein level. We saw that SERPINA3 cellular expression is actually upregulated upon conditioning with cerebrospinal fluid. We saw that by silencing SERPINA3, we can decrease GBM migration, proliferation, and also we can affect stem cell characteristics on GBM. And it's important because these stem cells in GBM are the main responsible ones for therapy resistance and tumor recurrence. And we saw that overexpression levels of endogenous SERPINA3 increases GBM migration and invasion. We also determined that by silencing SERPINA3, we can prolong survival in mice, as well as decrease proliferation index in these tumors. And we saw that there are higher levels of SERPINA3 in the cerebrospinal fluid that are derived from high-grade glioma patients. Next slide, please. And finally, I just want to thank my PI, Dr. Hugo Guerrero Casares, all the members in the Neurogenesis and Brain Tumors Laboratory at Mayo Clinic, Florida, as well as our collaborators that provided us with experimental resources or help us to collect the brain samples. And the most important, I want to thank the patients that donated the samples. Without them, this full project wouldn't be possible. And I want to thank the institutions that supported us academically and financially. Thank you very much.
Thanks so much. That was a great presentation. And with that, we'll move on to the next presenter, which is Aaron Long. Aaron will be presenting on evaluating clinical trial evidence for single-enantiomer drugs based on existing racemic drugs. Aaron?
Thank you, Dr. Shah. So I appreciate the opportunity to be a part of the Scholars Program and to talk about my project today. Next slide, please. So our study is an evaluation of clinical trial evidence on single-enantiomer drugs. So I want to take a second to talk about what those are. Enantiomers are molecules that are non-superimposable mirror images. An example of something that's a non-superimposable mirror image would be your hands or your feet. And then drugs that are composed of enantiomers are called chiral drugs. Chiral drugs are frequently manufactured as a racemic or 50-50 mixture of enantiomers, and a single enantiomer drug is just a drug that contains only one enantiomer and not both. Enantiomers can have different biological activities, so one enantiomer might theoretically have a greater therapeutic effect or fewer adverse events than the other enantiomer when used in a drug. Next slide, please. So while some drugs are originally introduced to the market as single enantiomer formulations, in other cases, a single enantiomer drug is introduced when there's already a racemic version on the market. This practice is known as chiral switching. So for as an example, the drug esomeprazole with the brand name Nexium is the S enantiomer of omeprazole, which is brand name Prilosec. And Prilosec is a 50-50 mixture of esomeprazole and aromeprazole. Drug manufacturers can receive a new drug approval for these single enantiomer drugs based on a racemic drug, and these are defined as derivative of a new formulation of an existing chemical entity, which would qualify for a 3-year period of market exclusivity, separate from the market exclusivity for the original racemic drug. So manufacturers can benefit from existing safety data from the racemic drug, but are not required to conduct any clinical trials directly comparing the new single enantiomer drug to the existing racemic drug, either before or after approval. The introduction of these single enantiomer drugs is often timed to coincide with the generic version of the racemic becoming available, making chiral switching a potential means for drug manufacturers to extend market exclusivity on an existing product. Next slide, please. So chiral switching imposes significant costs on the healthcare system. An estimated $17.7 billion could have been saved by Medicare from 2011 to 2017 if generic versions of racemic drugs were substituted for the newer single enantiomer versions. Furthermore, it's not clear if these newer, more expensive drugs offer superior efficacy or safety. A study in 2014 on 9 single enantiomer and racemic drug pairs found that pre-approval direct comparison trials were conducted for only 5 of those drugs, comparing them to their racemic precursor. And in only 3 cases did the authors of the trials actually present a direct statistical comparison between the drugs, meaning that in some cases, if the 2 drugs were in separate arms in the trial, no statistical comparison was made. So the evidence, it's not clear what evidence exists to compare these newer and older drugs and whether or not there's a significant therapeutic benefit to justify the additional cost. Next slide, please. So the objective of our study was to determine how often randomized controlled trials are conducted that directly compare a new single enantiomer drug to an existing racemic drug, both in pre and post-approval periods, and how often a statistically significant difference in efficacy or safety endpoints are found in the trials that were identified. We identified all single enantiomer drugs approved by the FDA up to 2019 that had an already marketed racemic precursor. And then we identified all randomized controlled trials reporting a direct statistical comparison between the single enantiomer drug and racemic drug in one of these pairs. Next slide, please. So we searched the drugs at FDA database to identify the drugs in our sample. You can see there are 15 drug pairs in the table with the single enantiomers on the left and the racemic precursors on the right, as well as the brand names of the drugs. You can see also in the leftmost column that single enantiomer drugs can be identified based on their prefix. So they'll have a prefix of dexo, levo, S, or R, which simply refers to which enantiomer is included in the drug. Next slide, please. Next, a research librarian designed a search strategy, and we conducted the equivalent of 15 systematic reviews, 1 for each pair of single enantiomer racemic drugs. Searches were conducted in databases for published papers and abstracts, and we also searched the clinicaltrials.gov registry of trials for any trials which were not published but had results reported online. In all, we identified 15,000 items for our title and abstract screen, and then screened 1,500 of these in full text. In the end, we identified 185 randomized controlled trials for our analysis. Next slide, please. So we characterized the results of these RCTs as favoring the single enantiomer drug, favoring the racemic drug, or favoring neither drug based on the statistical comparisons reported by the trial authors. Because trials are typically powered based on primary endpoints and secondary analyses are more susceptible to false positives, we distinguish for efficacy whether or not a difference was reported at the primary endpoint level or if it was reported for a secondary analysis. So for example, for a given randomized controlled trial, we might define it as favoring the single enantiomer at the secondary endpoint level for efficacy. Then for safety, we considered each trial separately from efficacy. So a given trial might favor one drug for safety and the other for efficacy. And we did not divide that based on primary or secondary endpoints because safety endpoints were rarely defined as primary in the studies. Next slide, please. So on this slide, you can see the summary statistics for the trials we identified. For the 15 drug pairs, there is a median of 2 RCTs conducted for each. And 30% of the trials reported an author affiliation or funding from pharmaceutical companies, while around 50% did not report their funding sources clearly. We just read within the text what was reported. We didn't do searches outside of what was reported in the text of the trials. Another thing to note is that in more than 2/3 of the trials, the single enantiomer drug was given at either a higher dose or the same dose as the racemic drug. Concerns have been raised in the literature that trials showing superiority of single enantiomer drugs have unequal dose comparisons because in theory, a single enantiomer drug contains twice the active ingredient per se as a racemic drug, given that it contains 100% of the active enantiomer. So having the two drugs at the same dose, for example, 10 milligrams may not be a fair comparison of the actual efficacy of the drugs. Finally, half the trials we identified did not define primary endpoints in their methods. Next slide, please. So on this slide, you can see a big picture representation of the findings from the trials we identified. The top figure is for efficacy and the bottom figure is for safety. You can see in the gray bars in both figures, those are the trials that favored neither drug based on the comparisons reported by trial authors. And you can see that for only 13% of trials was the single enantiomer drug favored for a predefined primary endpoint, while 14% of trials was the single enantiomer drug favored for safety. And then from the top to bottom on these, you can see in the top in green are the trials favoring the racemic drug, and then in blue are the trials favoring the single enantiomer drug. Next slide, please. So this slide gives a further breakdown of the efficacy comparisons for the 15 drugs. For 3 of the drugs, we did not identify any RCTs or randomized controlled trials that compared the single enantiomer and racemic. So you can see the 12 where we identified at least one trial. For only 6 of the 15 drugs, was there at least one RCT showing superiority of a single enantiomer drug for a primary endpoint. And of the 100, although we identified 185 trials, you can see that they're mostly clustered under a few of the comparisons. As I said before, the median number identified was 2 for each drug pair. And most notably for the comparison of levobupivacaine and bupivacaine, that accounted for more than half of our trials. One reason for this may be that this is a local anesthetic. It's used for a short period of time in a variety of procedures. So there just were like a large variety of studies that had been done that were of short duration with small enrollment. Next slide, please. So this slide similarly shows the safety comparisons. And for only 3 of the drug pairs, was there at least one randomized controlled trial showing superior safety for the single enantiomer drug. So again, you can see that in the gray bars here, most of the trials did not show a significant difference for safety between the 2 drugs. An important part of the argument for why single enantiomer drugs might be more beneficial is based in safety, because you're essentially cutting out a part of the dose which is not the active ingredient, or you're cutting out the enantiomer which is not having the desired therapeutic effect. So in terms of claims of the benefit of chiral switching, a lot of that would be in terms of safety. So for our analysis, we did not find very many randomized controlled trials showing a safety benefit of single enantiomer drugs. Next slide, please. So the first main takeaway of our study is that the single enantiomer drugs based on existing racemic drugs are rarely compared to the existing racemic drug. There was a median of only 2 randomized controlled trials per drug pair, and then for only 4 of the comparisons did we identify 5 or more trials comparing the drugs. And second, in the majority of the direct comparison trials, no difference was found for efficacy or for safety, and differences were especially rare when considering only the predefined primary study endpoints. Next slide, please. So just to briefly talk about the limitations of our study, First, we did not conduct any meta-analyses or risk of bias assessments for the trials. It was simply with 185 trials across 15 drugs and many different indications, we were not able to conduct more specific analyses. Rather, we wanted to give more of like a bird's eye view of what results were available for these drugs. And then we limited our search to randomized controlled trials. We did not include observational data, which could be illuminating for this topic, but we were interested in seeing how often trials, experimental trials are conducted. Next slide, please. So overall, newly marketed single-enantiomer drugs often do not prove to have superior efficacy or safety, although they do have greater cost to the healthcare system, and high-quality randomized controlled trials should be encouraged or required to assess the benefits of these individual new drug approvals and better inform physicians who are making, and healthcare payers who are making decisions about whether or not the new, more expensive drugs are worth the cost. Next slide, please. And if you're interested in reading more, we have a paper about this that just came out yesterday with the link there. So if you're interested in reading more, we have all of the data published there. Next slide, please. Yes, so thank you so much. I wanted to give a special thank you to my mentors for the project, Professor Joshua Wallach at the Yale School of Public Health, and then Dr. Ross, as well as to the CERSI Scholars Program for their support.
Thanks, Aaron, and congratulations on the publication. That's exciting to have it come out right on time. Great presentation. And then our last presentation of the session for this year is Mira Dodapkar. Mira is going to present about characteristics of clinical studies used for U.S. Food and Drug Administration supplemental indication approvals of drugs and biologics. Mira?
Hi, everyone. Thanks for having me. My name is Mira Dodapkar. I'm a second-year medical student at Yale, and today I'm going to be talking about my project, which was characterization of clinical studies used to support US FDA supplemental indication approvals of drugs and biologics from 2017 to 2019. And I just want to acknowledge my collaborators on this project before I get started, who are listed at the bottom. Next slide, please. So I know this audience is very well aware of these things, but just to provide a background for everyone, to receive US FDA approval for a new small molecular biologic drug, sponsors must submit adequate and well-controlled investigations to demonstrate the drug's safety and efficacy. And the FDA suggests that sponsors submit at least 2 trials demonstrating efficacy. These are known as pivotal efficacy trials. And the number and characteristics of pivotal efficacy trials supporting new drugs and biologic approvals varies widely. And this has been shown in previous work. Next page, please. And supplemental indication approvals are required when a sponsor seeks to add use in a new clinical indication or in a specific subpopulation for originally approved drugs. And supplemental indication approvals by the FDA for already marketed drugs are occurring in similar frequencies to approvals for novel therapeutics. In 2014, for example, the FDA approved 40 new supplemental indications as compared to 44 new drug approvals. And previous work has shown that use in supplemental indications is not uncommon. Certain therapeutics may even be prescribed more often for their supplemental indications for their initially approved indication. Next slide, please. And so the aim of this project was to characterize the pivotal trials that supported recent new supplemental indication approvals of drugs and biologics by the FDA from 2017 to 2019, and to compare the pivotal trials supporting supplemental indication approvals over this 3-year period to the pivotal trials that supported these same therapeutics' original indication approvals. Next slide, please. And so how we structured this project was a cross-sectional study characterizing the pivotal trials supporting supplemental indication approvals by the FDA from 2017 to 2019, as well as the pivotal trials that supported these therapeutics' original indication approvals. And we examined differences in trial characteristics and indication characteristics stratified by agent type, special regulatory pathway use, and therapeutic area, all of which were identified prior to data collection. And frequencies of use of randomization and double blinding among the trials in our sample were calculated excluding single-arm studies. So of note for the tables that I will present later in the talk. Next slide, please. So between January 2017 and December of 2019, the FDA approved 3,721 new SNDA and SBLAs. 3,200 were SNDAs and 521 were SBLAs, among which were 146 supplemental indication approvals for a new clinical indication for a total of 107 therapeutics. And among these 146 supplemental indication approvals, 99 were for pharmacologic drugs and 47 were for biologics. And 74 of these were approved using at least one special regulatory pathway. Most commonly, 61 were designated for priority review and 20 were designated for accelerated approval. And supplemental indication approvals were most often for the treatment of cancer, about 53% of the sample, and autoimmune and musculoskeletal disease, about 16% of the sample. Next slide, please. And so here you can see the summary statistics for our study for the first part. And next slide, please. And what I wanted to highlight here really is that between pharmacologics and biologics, there weren't many differences among randomization and double blind, but there was a difference among these two different types of supplemental indication approvals with regards to use of a placebo or active comparator arm, which you can see on the right side of the table, where pharmacologics were more likely than biologics to use a placebo or active comparator arm. Next slide, please. With regards to therapeutic area variation, what you can see is that cancer drug trials supporting in new indications in cancer were less likely to be double-blinded and less likely to use a placebo or active comparator arm. Next slide, please. Additionally, we assessed use of a clinical endpoint as well as trial duration and median number of patients overall and in the intervention arm of these trials. And what we found was that with regards to comparing pharmacologic versus biologics, the trials were very similar along these different parameters. Next slide, please. But with regards to variation between trial supporting indications in different therapeutic areas, we saw that the trial supporting indications in cancer were less likely than trial supporting indications in all other therapeutic areas to use a clinical endpoint as the primary efficacy endpoint, and they were longer in duration of note compared to trials in all other therapeutic areas. Next slide, please. Next slide, please. And these are more of the summary statistics which we discuss in our project. But what you can see here is that with regards to comparing the original indication approvals to the supplemental indication approvals for the same 107 therapeutics in our sample, trials supporting original indication approvals, they were more likely to be supported by at least 2 trials, and on average were supported by more trials. So you can see that on sort of the left side of the table. With regards to the number of patients that were at the indication level in the aggregated pivotal efficacy trials, there was similarity between both supplemental and original indication approvals. Next slide, please. With regards to the indication level characteristics of these trials, we found that pharmacologic and biologic supplemental new indication approvals were similar along these characteristics. Next slide, please. As well as between oncology and non-oncology indications. Next slide, please. Next slide, please. And so we also looked at other parameters at the indication level, and what you can see here is that original indication approvals were significantly more likely to be supported by at least 2 pivotal trials, but were otherwise similar along the other characteristics that we examined. Next slide, please. And what you can see is that pharmacologic and biologic supplemental new indication approvals were very similar along these characteristics when stratified at the indication level. Next slide, please. However, there were some differences between oncology and non-oncology supplemental new indication approvals when aggregated at the indication level, with oncology indications less likely to be supported by at least 1 double-blinded trial, 1 trial of at least 6 months duration, and 1 trial more likely to be supported by at least 1 trial of 6 months duration and 1 trial of 12 months duration, and less likely to have at least 1 trial with a clinical outcome as a primary efficacy endpoint. Next slide, please. So I just wanted to go over the limitations of our study. First being that we only assessed a 3-year sample of therapeutics approved by the FDA for supplemental new indication approvals. And we didn't evaluate whether the FDA considered other data regarding use of these therapeutics in other countries. And these may have informed the agency's efforts to evaluate supplemental new indication approval safety and efficacy. Next slide, please. And so the findings suggest that the evidentiary standards for supplemental new indication approvals varies across therapeutic areas, but that the FDA standard for approval is consistent between supplemental and original indications. We found very little difference between these two indications. And the evidentiary standard for supplemental indication approvals has important implications with regards to clinical care as well as regulatory decision making, and offers insight into whether the FDA follows a precedent set by therapeutics' original approval, or whether a lower or even a higher standard is used. Next slide, please. I know this audience is very familiar with this, but the 21st Century Cures Act, which was signed into law in December 2016, contained a provision to support the use of real-world evidence. And observational research may offer advantages with regards to time and resources, but as this audience is well aware, there's still some outstanding issues to resolve, including data definitions, establishing which data are fit for purpose, and establishing best practice with regards to things such as study registration. But there may be an opportunity to complement randomized trials to offer more information on product safety and efficacy in supplemental new indication approvals while we characterize appropriate uses of real-world evidence. Next slide, please. And I just want to acknowledge my collaborators and my mentors on this project, Dr. Ross, Dr. Shah, as well as the folks at CERSI Scholars Program, Tiffany Hu and Lara-Velazquesz, as well as Dr. Putumana, Dr. Jiang, and Dr. Downing. And I just wanted to share that this manuscript describing this work has been accepted for publication in JAMA Network Open.
Thank you, Mira. That was a great presentation. And that is our last presentation of the day. So now we have some time for Q&A. Please send any questions through the chat. I know there are some, and if we can get, yeah, all three of you are on camera now. So maybe Aaron, we'll start with you since we did receive a question in the chat related to your presentation. I don't know if you had a chance to see it, but the question is if there is, you know, that the driver for these trials is 3-year exclusivity. If there is no difference in safety and efficacy, what is the point of giving the exclusivity? So I'll let you answer that.
Thank you. I think that that's kind of the central question of all of this, is that drug manufacturers are not really incentivized to conduct direct comparison trials as long as they can conduct a trial comparing their new drug against a placebo and show it's more effective than placebo, and then market that drug as their older drug becomes generic. So I guess the question is there's whether or not there's the ability for the FDA to encourage drug manufacturers to conduct these trials, and then whether or not, based on the results of those trials, the approval process should change. As it is currently, drug manufacturers only need to show superiority against placebo. And I think as it stands now, since the original drug is well established, it already has recognition, and it gives drug manufacturers a chance to kind of hop patients from the old drug to the new drug as they come out with the newer the single-enantiomer drug. I hope that answered the question. I'm sorry, that was a little bit rambling.
Great, thanks, Aaron. I think while we wait to see if there's any other questions, I'll pose a question for Montserrat. Wanted to see what are the different real-world implications of the biomarker that you described today, and you know, a lot of great data with the biomarker, but where do you think the applications can be it in the clinical trials context or in real, you know, in the process of care delivery? Where do you think the applications for that biomarker might be?
I think that's a great question. I do believe our biomarker, or the molecule that we found, is not still a biomarker, but I believe it has a lot of future implications as a biomarker. Our data suggests that has strong implications in cancer development and malignancy, and now we are moving towards trying to find if there is a relationship between this biomarker and the early diagnosis of GBM. I think what we have to do now is to include more patients in our cohort, trying to see if there is actually different levels in the expression as in cancer development, because we don't know, we actually don't know if this SERPINA3 is gonna tell us there is cancer or not, like definitely it's gonna be a cutoff, or if it's gonna help us to see if it's gonna help us to detect earlier the development of GBM. So we have two different pathways that we can follow with this biomarker, but definitely I think we need to include more patients, start clinical trials if possible, which is kind of difficult because unfortunately human samples are very rare to get. And one of the main complications that we are having is the purity of these cerebrospinal fluid samples, because we are getting them while the neurosurgeon is resecting the tumor from the cavity, and some of these cerebrospinal fluid samples can get contaminated with blood, which is a problem for us. So there is definitely a challenge there to get a pure sample and to get as many as we need to really make a cutoff to say, okay, this works or not. But I think our data is really strong. And with many, in the following years, we can start clinical trials if everything goes as planned to really detect the use, if it's useful or not, our biomarker. Yeah.
Great, thank you so much. And then Mira, just a question for you, and this I'm maybe putting you on the spot a little bit, but you know, as you sort of alluded to the use of real-world evidence, and where do you see the context of where would you use real-world evidence versus where you might require clinical trial evidence in the context of SNDAs or
Yeah, I think that's a great question. I think one thing that our study really highlighted was that on average, supplemental new indication approvals were supported by fewer trials. And I think that there is a opportunity there to supplement those new indications with real world evidence, seeing as use, off-label use. Studies have suggested that off-label use is common, especially in different clinical scenarios, both inpatient and outpatient. So I think that there is an opportunity there to really leverage data that may be being generated in these specific scenarios. And I think our study highlights a specific situation where I think that it would be particularly helpful for establishing efficacy in these different indications. So I hope that answers your question.
No, that's a great answer. It's really helpful to sort of think about how to use what level of evidence and what context, especially for the supplemental applications. With that, I don't see any additional questions. So one thing I want to do is specifically thank Tiffany, who has been instrumental in pulling this seminar together for the day, and really appreciate all the time and effort Tiffany put into making this happen, and appreciate all the presenters. A great set of presentations today. And we personally, I think I'll speak for both Joe and myself, that we both like this format of having all the presentations in one day, and hopefully we'll repeat it again next year, maybe in collaboration with the other CERSIs as well. Thank you everyone for joining today, and have a great weekend.